All right, let's go on to biology. Biology chapter 1. Just give me 1 second. We're going to go right into mile down review sheets, the infamous mile down. So let me go show you guys chapter 25. All right. So the first chapter of biology is about the cell, right? So the cell is basically what makes up all of what living organisms are. And so I don't know why it's not on here, but they should be going over cell theory. Let's do it in light mode today. Just, oh, whoa, light mode's hurting my eyes now. Okay, let's go back. Cell theory. I don't know why it's not on here, but it should be. Because it's the first thing you learn about cells and biology. Let me check one more time. Yeah, no, it's not on here. So let's go over that cell theory first. Just give me 1 second. Okay, so cell theory is basically the core idea in biology that states that all living things are made up of cells, and cells are the basic units of life, and new cells come from older or pre-existing cells. So these are the 3 core rules of cell theory, and you should probably know them. They're introduced in a lot of biology courses. I don't know why it's not on here. Maybe it's not as high yield, right? Not everything is included in mile down, and in fact, there's been changes over the years with content, but overall these are still the main topics that you need to learn, right? So the first thing you need to learn, knowing that everything living is made up of cells, is you need to know the parts of what make up a cell. So the cell looks like this. I'm hoping you've taken some sort of introduction class and this is actually review, but yeah, this is what the the animal cell versus the plant cell. So in the cell, the nuclear region is the DNA in prokaryotes. So before we go into eukaryotes and prokaryotes, you need to know the difference between them a little bit. So prokaryotic cell versus eukaryotic cell. So as you can see, So as you can see in this picture, there are differences in them. Firstly, look at how they look, right? So this prokaryotic cell has a nucleoid, whereas this eukaryotic cell has a nucleus with a nucleolus inside of it. Okay, so that's the first thing to know, that in prokaryotes, it's called the nucleoid region where the DNA is, but then the nucleolus makes ribosomes, sits in the nucleus, but it has no membrane. So that's in your eukaryote, your nucleolus. So this and this are part of 2 different types of cells, right? So this is for the prokaryotes, and this is for the eukaryotes. I don't know why they didn't write that, but it's something you should know. Peroxisomes collect and break down material. So if you look at the diagram, I don't know why the peroxisomes aren't here, but if you don't know, they're called organelles of the cells. So just type that in, and you should get a diagram, cell organelles like this, that shows you. Oh, my computer's slow. That shows you peroxisome should be on here. Peroxisome. So they collect and break down material, rough ER and smooth ER. So kind of learn these together. ER is also known as endoplasmic reticulum. ER is just a short form. Rough ER accepts mRNA to make proteins, and the smooth ER is detox and make lipids. So that's the function of them. And where is the smooth endoplasmic reticulum? It's over there. Yeah, you also have rough endoplasmic reticulum, which is over here. It's even closer to the nucleus. Okay, the Golgi apparatus modifies and distributes proteins only in eukaryotes. So the Golgi apparatus is kind of like the shipping company of your body. It takes like vesicles and transports them around. It transports things around the cell. So vesicular transport. Let's just find it. So basically, there are 2 different views of how vesicular transport, or how the Golgi apparatus transports things. The first view, or like you could kind of say it's like kind of like a theory, vesicular transport versus cisternal maturation. So we'll go over those. So the difference between them is that I don't know if it's recording properly right now. But the difference between them is that the vesicular transport model is looking at the Golgi apparatus as the stable structure that doesn't really move, and the thing that moves is the vesicles or the cargo or the things that it's trying to transport. Like think of them as like bubbles of stuff, and inside of them there's stuff inside. And so COP2 means forward. So there's a protein complex which coats the vesicles traveling forward, and so then these vesicles traveling forward, they're coming from the rough endoplasmic reticulum to the cis Golgi. So the Golgi basically has like a cis side and a trans side. I should show a picture actually. So here we go. So there's a again a COP2 right over here, COP2, and basically this should mean forward, right? So this should mean anterograde, like that. So things are moving this way, right? These vesicles, these would be the vesicles that are moving this way. And they're moving from this rough endoplasmic reticulum, rough ER, right, to Golgi. So this is the Golgi. This side of the Golgi is called the cis side of the Golgi. So if you type in parts of Golgi apparatus, parts of Golgi, here's the trans face. Oops, here's the trans face, and here's the cis face. So we're pointing this way, right? So here's the cis face, and here's the trans face. So that's what they're referring to when they say cis Golgi. So this is a cis Golgi. And then COP1, sorry, is retrograde over here, retrograde. So retrograde is when the vesicles are moving in the reverse direction. Obviously, it's going this way now, reverse direction, from the Golgi, from this Golgi, back into the ER, endoplasmic reticulum. And that's basically the very basics of vesicular transport, right? Oh no, it wasn't recording the screen. Darn. Okay, now I have to explain it again. Damn. Okay, I guess I have to do it again. Alright, so unfortunately it was not recording the screen, so I have to explain it again with this. So here's the endoplasmic reticulum over here. And basically COP2 is where you have your over here is where you have your anterograde movement this way. And in this anterograde movement is also known as forward, right? Anterior it means like going forward, right? When you're going retro, it's like you're going backward. But anyway, we're on anterograde, and so these are the vesicles from the rough ER. And so the vesicles have a protein complex too. So that's what kind of what it refers to COP2. So these vesicles have that, and these vesicles are the bubbles with stuff. And for COP2, like I said, they're moving from the ER to the Golgi, and this side of the Golgi is called the cis face, cis, CIS. So there is a cis face and a trans face. So I'll go show you that. So as you can see, this is the cis face and this is the trans face, and right now we're talking about the cis face of the Golgi apparatus. So think of this side as the cis face, and the vesicles are traveling that way. Now, if we have COP1 over here, COP1 means a return back. So these vesicles have a protein complex 1, and they're moving this way in the reverse direction, retrograde. And so they're moving back to the endoplasmic reticulum, this area over here. Okay, so that's basically the basics of vesicular transport. All right, so now explaining the cis maturation model over here. The cis maturation model. This model is currently the favored view in biology, and instead of the vesicles doing the movement, so in vesicular model, the vesicles are moving from ER to Golgi, and even from Golgi to ER, retrograde, anterograde, retrograde movement, right? But in cis-ternal maturation model, we're looking at a model where instead of the vesicles or the little bubbles of stuff, you can also call them, instead of those jumping between these static, organelles. So yeah, the vesicular model saw these as just the Golgi staying in one place, ready to receive or send, and the ER ready to receive and send. But the cis-ternal maturation model is about this cisternal, like literally they call it cisternae, let's say cisternae, is able to physically move and mature from cis to trans. So new cis made, that happens when there's fusion of ER-derived vesicles, and it forms a brand new cis cisterna at the entry face. And so cis medial trans exit, the entire cisterna physically shifts forward through the stack, maturing over time. From cis medial trans Golgi network to exiting. So the vesicle stays inside of the maturing compartment, and until it matures, that's basically what happens. It just stays in that compartment, and vesicles travel in retrograde because the cisternae move forward carrying the cargo, or the vesicles, or the stuff, right? So the Golgi resident processing enzymes must be recycled backward. So vesicles, to explain it more simply, in the cisternal maturation model, you're looking at cisternae, which is compartments of the Golgi apparatus. So this Golgi apparatus here, it has compartments, like I mentioned, which were the cisternae, right? And so they physically move forward and they change over time rather than them staying in one place. So you can think of it as an analogy let's do an analogy, is that you can pretend that you have an assembly line conveyor belt where the workstation itself moves, carrying the product along with it. Or if you've ever been to an airport and seen, your conveyor belt, birth at the front happens at the cyst. So remember I told you what cyst face is, right? It's over here. Trans is on the other side. So birth happens how the vesicles coming from the ER fuse together to form a brand new cyst cisterna. That's what we're talking about, cyst. Cyst going from cyst to trans. It actually goes from cyst to medial to trans to add more complication to that maturation. So there's maturation happening, right? And the cargo stays put, or the vesicle stays put. The proteins inside do not jump from sac to sac in forward-moving vesicles. They simply ride inside the cisterna as it ages. So enzymes recycle backward or retrograde to change each compartment's function as it matures. Specific Golgi enzymes are packaged into retrograde vesicles COP1 and shipped backward to younger cisternae behind them. Exit at the back, trans. Once the cisterna reaches trans end, it breaks apart into secretory vessels that deliver the finished proteins to lysosomes, the plasma membrane, or outside of the cell. So basically, it matures within this Golgi apparatus, and then it's able to exit. And where does it exit? It exits at the trans side of the Golgi apparatus, where it could be delivered to other organelles in the cell. So that's basically what it is. Just go here, 1 second. So this is exactly what it says on your review sheet. Vesicles travel in, there's retrograde, new cyst is made, and the maturation happens in this order: cyst, medial, trans, and then exit. I would say vesicular transfer is easier to learn, but this one accounts for probably a lot of things scientists have found out over the years. Okay, peroxisomes collect and break down material. This is actually done twice already, so we didn't have to go over it. It's over here already at the top. So we already went over that. Centrioles are 9 groups of microtubules, and they pull chromosomes apart. So you should know that microtubules are like proteins, and you should know that centrioles are important for when we were talking about cell reproduction, or cell replication, sorry. So lysosomes, demo and recycling center made by the Golgi, so the Golgi apparatus, and they have a single membrane. So you'll notice that sometimes some organelles are made up of double membranes and some organelles are made up of single membranes, and it happens that the lysosomes are single membrane. Plasmids are only in prokaryotes here. In prokaryotes, they carry DNA not necessary for survival. Okay, now let's go into the cytoskeleton of the cell. So the cytoskeleton is made up of also microtubules, like similar to how the centrioles are made up of microtubules. The cytoskeleton is also made up of microtubules, and the microtubules are made up of tubulin. These are all proteins. So that's why everyone loves protein, and it's heavily overly marketed at this point. But protein is important. And your cytoskeleton is also made of microfilaments. And the protein is called actin. Intermediate filaments. Keratin is just kind of like one example, as well as desmin. Those are what your cytoskeleton is made up of. So cytoskeleton. I want to see better pictures of the parts. Yeah, this picture is good. So here you can see your microtubule with alpha tubulin and beta tubulin. You don't have to get too specific about that. Just know it's made up of tubulin. Here's your microfilament. Microfilament is made up of actin. Actin should remind you about muscles, but you won't learn that right now. And then here's your intermediate filament that looks like kind of rope-like, and its keratins coiled together in this one here. So keratin, you can think of hair and nails. That's how I usually think of it. Yeah. So you just have to memorize that. Tissues. So you have epithelial tissue, epithelial tissue or epithelia, and then connective tissue. Those are the two main. And so what is connective tissue? It involves your stroma, which is support extracellular matrix, bone, cartilage, tendon, and blood. So your blood is also a connective tissue, right? And then learning about epithelial tissue is basically you just have to memorize the layers of the epithelial tissue. And usually functional parts of organs in your body are made up of epithelial tissue, and those functional parts are also known as parenchyma. So here are the different layers of epithelial tissue. So you can have, let's see if I can find an easier diagram here. So you can have simple, you can have stratified, and then you can also have pseudostratified. And then as for the shapes, you can have squamous, cuboidal, or columnar. Columnar, remember, is just really tall, kind of. Cuboidal is obviously like a cube, and squamous is kind of like flat. So you have to know that. Cuboidal, cube shape, columnar, long and narrow, squamous, flat. And you can look at pictures to help you memorize that. Simple, stratified, pseudostratified. So yeah, this picture is perfect for you to memorize. Okay, now we're on bacteria. Bacteria, there's different types of bacteria. So let's talk about the shapes of bacteria first. So different shapes of bacteria could be bacilli, which look like rods, right? Or cocci, which look like circles or spears, and then spiral, which is spirilla shape of bacteria. So memorize those. And then the types. Obligate anaerobe requires O2. So the way you can memorize that is aerobic usually means aerobic system, or anything aerobic means I need oxygen, right? Anaerobic usually means I don't really need oxygen. But obligate, like obligatory, like it has to happen, or mandatory. Obligate reminds me of the word obligatory. So obligatory oxygen to me makes me memorize that it meant the bacteria really needs oxygen. Obligate anaerobe is when it dies in O2. Facultative, sorry, I can't really speak right now, it's kind of late. Anaerobe toggles between aerobic and anaerobic, right? So O2 and also not using O2, no O2. Aerotolerant anaerobe does not use O2 but tolerates it, right? So if it doesn't use O2, then what is it using? Anaerobic system. An anaerobic system. That's why it's still an anaerobe, but it's aerotolerant. So it will be able to tolerate the O2. It won't die like the obligate anaerobe. It just dies. It has to be anaerobic and not use O2. It'll just die. Okay, anyway, gram positive is purple. So this is what happens when you put the bacteria to slides and you're looking at it. Gram negative is pink red. So thick. This is because thick peptidoglycan and lipoic acid cell wall of a gram positive, and a gram negative is because of the thin cell wall and outer membrane. So thin, thick, gram positive, thick cell wall, gram negative bacteria, thin cell wall, or thin cell wall and outer membrane. Okay, so now we're on genetic recombination. So how does bacteria end up reproducing in a way, and what does it do? How does it interact and stuff like that? So for bacteria, there's transformation, gets genetic information from the environment. That actually, yeah, gets genetic information from the environment. Conjugation is like bacterial sex, basically transfer of genetic info via conjugation bridge, right? So if you're so the F plus goes to F minus, so it has to be an F plus bacteria with an F minus bacteria. So if we go conjugation bacteria, actually this is a good summary. So you have transformation, transduction, conjugation, there's a sex plus, and transfection. Transfection isn't really on the review at all, but these three were. So if you look at this one for conjugation, you have one donor and one recipient, right? And one of them has the pilus. I believe the one that has the pilus is the F, I think it's F plus. So bacterial conjugation is direct transfer of genetic material, requires a donor cell with a fertility factor F plus. So your F plus needs to pair with an F minus for the recipient. Functioning as a primary way, share traits like antibiotic resistance. So yeah. Or HFR to resistant. Actually, to resistant bacteria. So basically, this is high frequency recombination. Bacteria's name has fertility plasmid integrated into the main chromosome. Alright, so transduction is a transfer using bacteriophage. Transposons is genetic info that can insert or remove themselves. So transposons, right? So transposons, you'll learn a little bit more when you go into DNA and cell replication. But for now, just know that it's basically any genetic info that can insert and remove themselves. And that's not really specific to only bacteria, right? So a conjugation is, yeah. So now we can go into the differences between a eukaryote and a prokaryote. So I already showed you a diagram, but here are the high yield differences. So in a eukaryote, you have your electron transport chain, which is in your mitochondria. But the prokaryote, it's in the cell membrane. So your electron transport chain is involved in cellular respiration. If you don't know what that is, probably have to review that as well. But for now, just know ETC is your electron transport chain. Large ribosomes versus small ribosomes reproduce via mitosis, so eukaryotes do that. But prokaryotes reproduce via binary fission, which is kind of like mitosis, but it's bacterial mitosis. Plasmids may carry DNA materials, and they have virulence factors, plasmids that integrate into genome or episomes. So we need to define virulence factors. So I'll do it in a very definitive manner. So virulence factors are traits or molecules produced by pathogens that allow them to colonize a host, evade the system, and cause a disease. So virulence factors is basically like molecules and physical structure traits produced by pathogens, so bad things like bacteria, viruses, fungi, and parasites that allow them to affect a host. And multiply and cause disease. Virulence factors is one of the ways that even though you might have a strong immune system, you might get sick anyway, basically, from the cold or, yeah. Okay, let's go into episomes too. Episomes. So episomes are a piece of genetic material that can replicate either independently of the host cell's main chromosomes or by integrating directly into it. Okay, so now the important part is about viruses. So viruses are basically, we've heard of them before. The bacteriophage, it is an example of a virus, but it's also a high yield virus. That's why it's probably here as a picture. And you should know the bacteriophage has a head with DNA and RNA, collar, sheath, base plate, tail fibers. I believe there's even more stuff to remember, but this is the high yield stuff, right? And then you have a capsid, which is your protein coat, envelope, and a virion, individual virus particles. So this is also what the bacteriophage has as well. Whoops, it's so bright. And bacteriophage is a bacteria virus. The tail sheath injects. The tail sheath is here. So a lot of these parts are not even on this diagram, so I'll show you a better diagram. Parts. Here's the collar. Yeah, this is pretty good. So here's your protein coat, the DNA, right? They say protein coat is your capsid. Then you have your envelope with some lipids. I'm not sure if it's showing envelope. It's not there. A virion, individual virus part of bacteriophage, tail sheath. So your tail sheath is here, or yeah, here. Yeah, this is your sheath. These are the fibers, but the sheath is here. This is what injects. So you could see this happening, injecting in, right? And then virogenome may be DNA or RNA, single or double-stranded. If it's single-stranded, you get positive sense or negative sense. So if you get single-stranded positive, it can be translated by the host cell. But if it's negative, it can't be directly translated by the host cell. It's like it has to go through a little bit more. So it has to have an RNA replicase, which an enzyme must synthesize, like a protein, has to synthesize a complementary strand, which then can be translated. So it has to do an extra step, which is synthesizing a complementary strand. And then a retrovirus, is made of has single-stranded RNA. So you need an enzyme reverse transcriptase for the retrovirus to make DNA. Yeah, and then bacteriophage and life cycles, there's lytic and lysogenic. So virions made until cell lysis. In other words, the cell like basically explodes. Like when a bacteriophage enters lytic cycle, you need to know the lytic cycle versus lysogenic. So I'll show that. So yeah, if you enter the lytic cycle, basically it keeps going. Oh, that's on the sun. Keeps going, keeps replicating until the cell basically explodes and dies. But in the lysogenic cycle, it's basically the bacteria is in there. It's in there, and it has its DNA in there, but it's like stealthily hidden. It's like hidden in there. Nothing knows until it may go into the lytic cycle, maybe, right? And then that will cause the explosion. So your virus integrates into genome as a provirus or a prophage. It goes dormant. That's what I was talking about, where it's kind of like hiding. No one really knows it's there, so it's dormant until a stress activates it. So until some sort of stress activates it, it may enter the lytic cycle, and then the cell could have the potential to explode or lyse. And then miscellaneous, this is probably mild down, didn't know where to put this. Prions are infectious proteins. They trigger misfolding of the alpha helical and beta pleated sheets. Alpha helical and beta plated sheets is in reference to protein structure. So protein structure you learn in biochemistry. So if you don't really know that, you can review that as well. Leads to less solubility, and then viroid plant pathogens. I should probably go over just the basic of what is a pathogen, because that's like everywhere. A pathogen is any biological agent or microorganism, such as a virus, bacterium, fungus, parasite, that can produce disease and sickness in a host organism. So there are viruses, right? There's bacteria, fungi, parasites. There's also a lot of other things more than that, but these are some good examples. Okay, we'll stop there for now. Okay, so now we're gonna continue on with biology 2 about reproduction. This is chapter 2 on reproduction. So reproduction in terms of biology is not just about reproduction of, usually we think of reproduction as reproduction of animals, like this animal gave birth, such as your mother probably gave birth to you, right? Something like that. But reproduction also, at least in this chapter, mostly looks at the reproduction of cells and how does that reproduction work? And honestly, the primary focus of reproduction actually, yes, is the female reproduction. It's also going to be focused on the fetus. So we're going to first go over how do cells reproduce, because like we said in chapter 1, all living organisms are made up of cells. And in order to, make another, baby, or living organism, then we're going to need cells. And we're going to need cells to be able to reproduce in some way, shape, or form. So let's start off with the cell cycle. Okay, so your cell cycle, it's like a life cycle, it's kind of like the seasons of the year, for example, spring, then winter, and, summer, fall, right? So you have the cycle that happens, and it repeats every year, like the seasons. And so your cell cycle goes in these stages. So first is G1. I'm not sure if it's easier to see in light mode. It's a little bit easier to see in light mode, but it's hurting my eyes a little because I'm doing this at night. But as you see, G1 is first, so G1 is the first stage. And in this stage, your cell is making mRNA, which is ribonucleic acid, messenger ribonucleic acid to be more specific, and then proteins to prep for mitosis. So mitosis is the way in which your cell can divide and replicate itself. So in essentially, it's kind of like its own reproduction method, right? And then you hit G0. So what G0 is that sometimes the cell can decide and say, you know what, we're not dividing today, we're just going to rest. And in G0, is when the cell determines it does not want to divide or do anything. It's just going to stay in a resting state almost. A G1 checkpoint is basically you have a G1 checkpoint and you also have a G2 checkpoint. And in these checkpoints, it's kind of like your cell is checking literally what's going on, what's happening, and before it continues any further, right, with the process or with the cell cycle. So in the G1 checkpoint, the cell decides if it should divide, and this P53 is in charge. So P53, I think, is like a gene. Let me go search that. Okay, sorry. So here is, oops, why is it not? Here's the, I don't know why it wasn't going up. But as you can see, the P53 protein acts as a crucial tumor suppressor and the guardian of the genome at the G1. So it's not a gene, but it is a protein, right? When a cell encounters DNA damage during the G1 phase, P53 stabilizes, accumulates, acting as a transcription factor to halt the cell cycle, meaning it stops the cell cycle whenever it encounters DNA damage during the G1 phase. So it's able to stop this, and it turns on the gene for P21, and that's a little bit out of scope, so you don't really need to know that. But basically, P53 is in charge. All right. And then in S stage, this is where your DNA gets replicated. Your DNA is being replicated, and G2 is when the cell grows even more, and it makes organelles. G2, your cell's checking for its own size and organelles, make sure it's all grown out. And then M stage is finally mitosis. So we reach finally to M, which is mitosis and cytokinesis. Cytokinesis is part of, to me, how I learned it is cytokinesis is basically part of mitosis, and it's part of the telophase of mitosis, which is the last stage of mitosis. Or you could just think of it the very last stage of everything after PMAT. So I'm going to then explain mitosis and cytokinesis because that seems like the most logical way to go about it. So mitosis is basically like I said before, how your cell likes to reproduce. And so PMAT is an acronym that you can use to help you memorize these stages, and it stands for prophase, metaphase, anaphase, and telophase. So you can use PMAT to help you memorize that. Ploidy of 2N throughout. I'm going to explain this afterward. So it's not too distracting. So basically in prophase is where your DNA is going to condense, and centrioles migrate. So we learned about centrioles in chapter 1. Migrate to opposite poles. So pretend this is your cell. Your centrioles are migrating to opposite poles, right? Opposite sides, in other words. You can put sides. And microtubules form, and your nuclear envelope disappears. So this stuff happens in prophase. And then in metaphase, meet in the middle. What does it mean, meet in the middle? So some people like to remember metaphase and its function by saying meet in the middle, and the reason why, or M for middle, and the reason why is because this is where your chromosomes, literally, quite literally, they meet in the middle, like this, like this, like this, like this, like this. And then in anaphase, people like to memorize anaphase by remembering A for apart, anaphase for apart. And what that means is your chromosomes, so this was M, right? In your C phase, I should draw it the other way so it doesn't look confusing. Sorry, not the best artist. Cytokinesis. So there's pulling apart. So these have like, pretend these have like a graph on these guys. So they're going to be pulled apart, away from each other, right? All of these chromosomes are away from each other like this. And this starts to divide it. It starts to divide here. So A for anaphase, apart, sister chromatids separate and they move to opposite poles. So here's your chromosome, right? And then we'll talk about chromosomes versus chromatids in a sec, and we'll also talk about the ploidy, because these are all terminologies which basically describe chromosomes in a sense, so I'll explain that later. And then in telophase is when chromosomes decondense and your nuclear membrane forms, and then cytokinesis occurs. So the way you can think about this nuclear membrane, I think of it as like, oh, your DNA thing is opening and closing. I don't know, but yeah, cytokinesis occurs. So cytokinesis is when all the splitting happens, and I wanted to add that cytokinesis is the final step where a single eukaryote splits its cytoplasm into two separate cells. So cytokinesis is basically the official splitting, right? So they're officially split at that point. So that's what cytokinesis is. Now, one thing in, I think, chapter 1, they didn't really explain too well, is that, yes, your DNA is found in your nucleus, but more specifically, or in the eukaryotic cell, right? But more specifically, this is a chromosome, as you're looking at it. It looks like an X. And one of them, there are actually two sister chromatids that are crossing over each other. And there's actually more about, like, chromosome anatomy to know. This one's pretty good. So your chromatid is just one of them, and then your chromosome is this whole thing, right? Here's another one. You don't have to know P arm, Q arm. Centromere might be good to memorize. Chromatid is definitely good to memorize. Telomeres are important to memorize. And you have to know, basically, chromosomes are your DNA. So yeah. And humans have 23 pairs of chromosomes. And they say that your 23rd determines your biological sex. All right, so another thing, oh yeah, I forgot to talk about ploidy, one sec. All right, so you learned in class never to learn through Wikipedia, but I verified that it's saying pretty okay stuff, okay? So ploidy is a number of sets of chromosomes in a cell, and hence the number of possible alleles for blah, blah, blah. So if we go back, yeah, ploidy is a number of complete sets of chromosomes in a cell. So common types, there's a haploid, which is one set of chromosomes found in the human sperm and egg cells. Okay, so for sperm and egg cells, it's a haploid. For diploid, you have two complete sets of chromosomes, right? One set from each parent found in standard human body cells. And then this triploid and polyloid you won't find in humans, by the way. So you only find haploid or diploid, okay? And this would be haploid and diploid. Knowing what that means is important for your MCAT test, right? So I wanted to clarify that the sperm and egg cells are also known as gametes or sex cells. Diploid cells are regular body cells, and those are also known as somatic cells. So that's important to know too. Here I found another resource where someone explained it really well, and they said, the way I remember in high school, diploid has double, haploid has half, and if you can remember that haploid equals half a diploid, then you know how many chromosome sets are in one based on the other. So 2H is equal to D, half of D is equal to H, because haploid is half of the diploid, right? So half of D is equal to H for some simple math. If D equals 10, then H will equal 5, right? Because haploid is half of D. If H is equal to 12, then D is double of half. So 12 times 2 is 24. If you're given haploid, multiply it by 2 to get diploid. If you're given diploid, divide by 2. I hope it helps. So there you go, guys. All right, so now we're going to go into probably meiosis, because meiosis builds on mitosis. So meiosis is basically the same process, but it's done 2 times, as you see here. Non-disjunction, when the sister chromatids don't separate properly during anaphase, results in aneuploidy. So this is just something that can happen. So let's go over the normal process. So the first thing that happens is your prophase, but you'll notice these Roman numerals here. So you have prophase 1, metaphase 1, anaphase 1, telophase 1, and then you have prophase 2, metaphase 2, anaphase 2, telophase 2. So this is PMAT number 2, this is PMAT number 1. So PMAT happens twice, unlike mitosis, which only happens 1 time, right? So Whoops, too much highlighter everywhere. Whoops. So chromosomes condense again. And then also something different is that crossing over occurs. Crossing over occurs. So what is crossing over? What is crossing over? So I drew this diagram actually kind of poorly for mitosis because crossing over, this is more it looks like meiosis actually. Crossing over. Here we go. So this is what happens in meiosis, and this is how you get pairs that happen together. So in mitosis, versus meiosis, in mitosis you get a single file line, which I didn't draw correctly the first time, but I just wanted to draw it that way to show the splitting. But basically, I'm going to draw it again. I'm going to draw it on the paint so it's easier to see. But basically, let's say I have, let's do meiosis first, or sorry, mitosis first. So we know mitosis is PMAT times 1, right? So let's now draw a cell. This is my cell, and I have my chromosomes that are lining up. They're condensing, so they already did P phase, preparing phase is how I like to remember it, prep phase. And then they're already lined up in the middle, so metaphase, right? So in the middle, M for middle, if you want to memorize it that way, and then A for apart. So let's do the apart part first. We've already drawn the centrioles into microtubules, but in the prep phase, they should have established some of that here, right? And then so they're connecting like this. So they're connecting like that, like, I don't know, like claws, I don't know. And then what they're going to do is they're going to pull the sister chromatids apart. So let's go over chromosome again. Chromosome versus chromatid. So remember, your chromosome looks like, whoops, my bad. So your chromosome looks like this. This is your, whoops, okay, I don't want to draw a circle or else it's going to look confusing. Here's your chromosome, and then your chromatid is just one half of it. And then the other one, see, it's hard to see because they're interlocked, but you can think of it as two chopsticks making an X, and one of them's on top of the other one, and there's two chromatids, right? So a chromosome, just to give an analogy, is like two sets of chopsticks crossing over, right? At the central mirror, I think it was called, or central mirror, I think, right? It's crossing over at this point here, and then now it's pulling apart in meiosis this way. It's pulling it toward these poles. This one's going this way, and this one's going in the opposite direction. So you pull two sides of it over, which will give you, if I pull this, I get one on this side and one on this side, and they split. So what happens, telophase is where you get chromosomes decondense, nuclear and cytokinesis occurs, right? So in telophase, you basically get two cells finally made in cytokinesis, right? So now the two cells have been ripped apart, split with the single, so these are the single chromosome things, right? So I put 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, and the other half of them, 2, 3, 5, 6, right? So now it's split. So these, what happened to them, remember, is that they split in half. One of them pulled this way and one of them pulled that way. Okay, so that's mitosis. Now let's go into meiosis. So meiosis, again, was PMAT times 2. So the difference in meiosis that I wanted to explain is the crossing over. So I'll draw this quickly, is that you have this crossing over in prophase 1. So it says homologous chromosomes form bivalence, crossing over occurs. Essentially, they are just going to connect together in pairs. They're also called tetrads here. Tetrads align crossing over. So tetrads are two pairs of sister chromatids. So they cross over each other. So why can they be in this lineup like this? It's because they're not supposed to be. The way I drew it wasn't very precise. I'm not drawing it precise right now. But if I were to draw it more precisely, there's this crossing over that happens between them. Essentially, these are actually linked, right? So they're linked together. So these are physically linked together. These ones as well, they're linked together. Crossing over occurs, that's what it means. And they form what we call tetrads, which are basically these pairs. Tetrads is just a name for that. And now that one happens, and then spindle fibers from opposing centrosomes, so same thing about the centrosomes here. They're opposing on different sides, connect by valence at centromeres, and align them along the middle of the cell. So they're connected. And they get aligned in the middle of the cell. I already drew them in the middle. So, yep, they're connected. And now what happens is, Homologous pairs move to opposite poles of the cell during anaphase. This is disjunction and accounts for loss segregation. So we'll go over that in a second. I just want to go through most of this first. So yes, they are moving to opposite sides of the cell. So they should begin to look more like this, moving more towards the opposite side. Just say I drew enough. 1, 2, 3, 4, 5, 6. I always draw 6. Wow, 1, 2, 3, 4, 5, 6. Okay. There doesn't have to be 6, by the way. There's probably remember the numbers I told you were in human cells and stuff? Yeah, it's not just like 6. Yeah, no. Anyway, so these are pulling apart to opposite sides, and they're going closer to their sides here. And now, chromosomes decondense, nuclear membrane may reform, cell divide, cytokinesis, and it forms 2 haploid daughter cells that are unequal in sizes. So if I could just expand this board a little bit downward and just show you that basically this will start to divide into 2. Now you have 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And they said they were haploid. Yes, haploid daughter cells. These are called daughter cells for obvious reasons, right? Okay, so now we went over that. I'm going to finish over prophase, metaphase, anaphase, telophase, and then we'll go back to fill in some extra details such as disjunction, which we haven't learned yet or haven't reviewed with yet, and law segregation soon. But yeah, let's continue to PMAT times 2 now. So prophase 2 is when chromosomes condense, blah, 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 and basically centrosomes move to opposite poles, perpendicular to before, same thing as before. Okay, prophase 2 is the same thing. Metaphase 2 is spinofibers, blah, blah, blah. They attach and you align them in the middle of the cell. So same thing, exactly the same thing, right? Anaphase, spinofibers contract and separate. This image is probably better. Yeah, so they contract, separate the sister chromatids. Chromatids are now called chromosomes, move to opposite pole. So let's just show those. So what happens here is, if I can just make this larger, it's very interesting because basically it happens again, PMAT. So this will split and this will split. But let me show you the process of the splitting. So the central sums and stuff, pulling apart, pulling, lots of pulling happening. Lots of pulling. Even on this one, the same thing is happening, pulling, right? Pulling them apart, and then they start to get pulled apart, and now it looks like mitosis. This looks like mitosis. So meiosis number 2 looks like mitosis. Because look, now we have the single file line just like in mitosis. And now the same thing is going to happen just like in mitosis. 1, 2, 3, 4, 5, 6. 2, 3, 4, 5, 6. Let's split the cell, split it up, and then you have more of these guys. This one is done splitting, should be done splitting, and then you get cytokinesis, where it splits fully into their individual cells, and you get 1, 2, 3, 4, 5, 6. 1, 6. There you go. So they split apart. No, it was done pulling, my bad. They split apart, and now you have 4 haploid daughter cells. So these are now 4 haploid daughter cells. You don't see these 2, but this one is here. Whoops. This one is here. These 2 are here too. So these, if it goes through the same thing on this side, both of them go through it, right? So they split. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And then it's fully split, right? So, and then you get 4 cells. So here's 1, here's 2, here's 3, here's 4. And those are your 4 haploid daughter cells. All right, so now we're going to move back and explain a little bit about disjunction and law of segregation. All right, so basically, just to help explain disjunction, disjunction is the physical separation of chromosomes during cell division. It's just a way to say that the chromosomes are physically separating. While the law of segregation is a genetic principle, right? So you learn law of segregation again in the genetics chapter of biology, so you don't have to worry too much right now. But basically, it's a principle that describes how allele pairs split into separate gametes. So how does your DNA split? How does that split into separate gametes? Well, I don't want to use DNA just randomly, but how do we get our gametes, right? And so disjunction, in genetics, a junction is a connection, right? So when I have a disjunction, it just means the normal separation of genetic material. Disjunction, the separation, right? I just told you that disjunction is a physical separation of chromosomes during cell division. Yeah. So that's what it is during anaphase. Remember, anaphase was the pulling apart? Yeah, that's disjunction. Again, physical separation. So when do you get disjunction? You get disjunction in meiosis 1, right? So where's meiosis 1? Here, and specifically in anaphase. Whoops, that's why they mentioned it there. Why can I undo it? Right here. And then you get meiosis 2 and mitosis. So it happens in mitosis as well, right? Anaphase. And then the genetic rule is Mendel's law of segregation. So this is Mendel's. I didn't really want to introduce Mendel until I did like more in depth about Mendel, but we'll do a little bit for now because I want to explain how does disjunction account for law of segregation. So Mendel's first law says that every diploid organism carries 2 alleles for a given gene. So you can think of an allele as a type of your gene type. Here's the actual definition though, so you don't think I'm just saying stuff. An allele is an alternative version or variant of a gene at a specific location of a chromosome. So the way I like to think of it is like, I don't know, brown eyes, blue eyes, right? So that's how I like to think of alleles. So yeah, different variants of a gene essentially is alleles. And so every diploid organism carries 2 alleles, and these 2 alleles separate or segregate during meiosis so that each gamete receives 1 allele, right? So the connection is that disjunction during anaphase 1 is the exact physical mechanism behind the law of segregation. So suppose, so I don't want to go too much in detail, but yes, in anaphase 1, you have like homologous, and anaphase 2, you get separation, right? Disjunction is the physical act of pulling the chromosomes apart. So it counts for law of segregation, which is the genetic outcome where 2 alleles separate, so each gamete only gets 1, right? All right. So remember earlier how I mentioned somatic cells versus gametes, or these cells, right? I don't want to get blocked by YouTube by putting that in fully. I don't mean the other way. I mean like the terminology they usually use. That's how they usually use it. But basically, the law of segregation and separation of homologous chromosome pairs applies specifically to only meiosis. That's why you didn't see this annotation about law of segregation in mitosis. So basically, remember I was talking about somatic cells and gametes and how they're different in being diploid versus haploid, right? Haploid. So again, remember which one was diploid, right? The result of 2 identical diploid cells, that is in mitosis. So these are your cell types, right? Cell types, right? And these are the ploidy that the cell could have or be, whatever. So somatic cells use mitosis, right? So this is a broader overview of what's happening, mitosis, right? And then these are like your body cells, in other words. Your reproductive cells, right, are using meiosis. They separate into 4 daughter cells, right? And they result in you having 4 genetically, why did I draw that far in haploid gametes. So you get a lot of what we call genetic diversity. And you'll learn more about that in the genetics chapter, but not right now. But yeah. Oh wait, you couldn't see any of that. Could you see any of that? Wait a damn minute. Could you see any of that? Yeah, you must have saw that. Okay, never mind. I don't know what I'm thinking right now. Okay. So yeah, and the pictures over here, it's better in light mode probably to look at it. I find the picture is kind of not HD, so maybe find a good picture on Google of my mitosis and meiosis. Okay, now we're going into growth signals. I'm just gonna take some water first, and then because my throat hurts now. All right, so I thought I'd explain this in more of an analogy because it would be much easier for people to learn with real life analogies that make it more simple to learn something. So it makes it more easier to digest. Yeah. So let's start with growth signals, right? So let's think of your cell as like a car that wants to drive forward. So what is its forward movement? What is the natural progression of the cell? What does it want to do? Okay, well, we just went over the cell cycle, right? So it wants to eventually hit mitosis where it's dividing. Okay, so the cell wants to drive forward and divide, but it's parked on a hill with a tight handbrake, okay? So it has some control over its growth in a way. So this E to F, so we have a few characters here, okay? So let's say you have the car. I'm not really good at drawing, but let's say you have the car. Here is my car. Whatever has windows and stuff. Kind of looks funny a little bit. And E2F. Let's introduce some of the characters. So E2F, RB, cyclin plus CDK, and phosphate. So these are some of the characters that we're going to introduce now. So we have E2F, which is the gas pedal, a protein that desperately wants to push the gas pedal and copy the DNA. So we want to drive forward into more growth, more cell growth. That's what your cell wants to do, right? So your E2F is kind of like that gas pedal. So pretend you have your driver here driving and they're stepping on the gas pedal. That's their hands on the wheels and they're stepping on E2F. So there's E2F, right? And then you have RB. So RB is like the handbrake, a protein that grabs onto E2F and keeps it locked down so the cell cannot divide. Okay, so you have the special handbrake on the side. Let's say this is your handbrake, and that would be RB. And on the back end, your handbrake is gonna stop E2F, stops it. Okay, so it's gonna stop what we call cell growth too, right? In doing that. Now, cyclin plus CDK, the handbrake the hand that releases the brake. So a pair of helper proteins that team up when the body says grow. So who is this helping? This is helping E2. Whoops. These two are helping E2F, right? So they're going to, what are they going to do? They're going to team up together, both cyclin and CDK, that release this RB. So it doesn't act anymore. No more acting. No more onto E2F. Now, phosphate is a sticky note, and it's a small chemical group that turns proteins off or changes their shape. So let's go into scenario 1. So let's say your cell is driving into more cell growth, right? And the stoplight right now is green. Okay, so it's on go mode, right? So growth signal arrives, the cell gets a message that it needs to multiply. So let's go back to the review sheet that was there. CDK plus cyclin create a complex. Again, they make a team together. They work as a team. And then phosphorylate RB to RB plus P. RB changes shape and releases E2F. Remember, E2F is driving that signal forward, right, to grow, and then cell division continues as a result of that. And then negative growth signals is the blocking. So that would be your red light, right? So red light is when CDK inhibitors block phosphorylation of RB, so E2F stays attached and your cell cycle just halts. So it stops. So let's go again and review that. So you have this green light, go. You want to do more cellular growth naturally, right? Or that's at least what your cell wants to do. And so in the red light, your growth signals arrive. The cell gets a message that needs to multiply, and so cyclin meets up with CDK and it forms a team, right? So these two work together in a team. Now, CDK tags RB. The cyclin CDK sticks a phosphate onto RB. So they take the sticky note. Remember we said that phosphate was like a sticky note. So these two players are going to take phosphate and stick it onto RB. Okay, now they have stuck the phosphate onto RB. RB, let's go. When RB gets tagged with the phosphate, right? So we stuck the phosphate, right? It will change shape and accidentally drops E2F. So RB changes its shape. Or if you learned it in other courses, maybe changes conformation. I'll just put changes shape for simplicity. So it changes its shape. And now that RB has changed its shape, it's no longer having control over E2F. And now E2F is free. It's free. There's no handbrake that's on E2F right now telling it, you need to stop. It's free now. So no more blockage, right? So we could delete this blockage. No blockage here. RB can go away from the picture over here. And now E2F is free to drive, its forward growth, cell growth. So that's a green light. Now let's talk about what happens if you have a red light. Let's see if we have a little bit bigger here. If you have a red light. So this means negative, no cell growth. You need cell growth to stop. So what happens then? Well, you can guess that RB has a big play into it. So trouble is detected and the cell notices something wrong, like damaged DNA. So the reason why your cell wouldn't want to grow is if it had damaged DNA and wouldn't want to replicate. It's like cancer, right? It doesn't want to replicate bad DNA. So it's taking care of you. So bad DNA is one of the reasons, right? So that your cell does not want to grow. And so an inhibitor steps in and blocks cyclin and CDK from working together, and RB stays locked. So this one stays in its place, trapping E2F. So E2F is not free, right? Cyclin and CDK are also, what are they doing? They're not even working together because an inhibitor has blocked them. Inhibitor has blocked them from working together as well. So E2F stays trapped. RB holds tightly onto E2F. And so your handbrake, essentially, remember RB was your handbrake, it stops this whole process of cell growth, and it halts it. It stops it. With E2F locked up, the cell cannot push the gas and safely pauses until damage is fixed. All right, so I hope that analogy helps. Oh, whoops, what happened? Now I can't see anything. So that's how I can explain growth signals in a better way, I guess. Now we can go into sex chromosomes. So your sex is determined by the 23rd pair of chromosomes. I already introduced earlier. I don't even know why it's not really on the review sheet. That your eukaryote cells have 23 pairs of chromosomes, and your 23rd one is your chromosome that determines your biological sex. So XX would mean female, and XY would mean male. Your X-linked disorders, males express, females can be carriers. So in X-linked disorders, something to note about them is that it's more common to see like an X-linked disorder than it is to see a Y-linked disorder. So why is it more common to see X-linked than Y-linked disorder? All right, so here's the thing. X-linked disorders are much more common than Y-linked disorders because the X chromosome contains over 800 active genes essential for normal function and survival, while the Y chromosome has fewer genes, mostly responsible for only your male sex determination and fertility. Right, so that means your X-linked disorders are going to be more common. Your Y chromosome has very little genetic info, as I just Googled for you guys. It has little genetic info, and it's related to the gene called SRY gene. The way some people like to memorize this is sorry, you're a male. So SRY means sorry, looks like sorry, so it's easy to memorize. And then the phrase sorry, you're a male correlates the SRY gene to male, right? So that helps some people memorize. And now we're going to go into the male reproductive system. This is mostly memory. So semen is made of sperm and seminal fluid. Your the male bulbourethral glands makes viscous fluid to clean out urethra. So that's a part of, I recommend searching these parts in terms of anatomy pictures. I won't show the pictures, but you by now should have seen them before. And if you haven't, you have to go look at the pictures. So you need to know urethra, bulbourethral glands, semen, what it is made up of. Seminal vesicles and prostate glands make alkaline fluid to help sperm survive acidic environment of the female reproductive tract. So you should also know by now that part of reproduction is that somehow the sperm needs to enter the female body and interact with the egg, right? So in that environment where the sperm is inside of the female reproductive tract, it's going to be acidic. So for the sperm to survive that acidic environment, there needs to be some type of fluid that helps it. And if you don't know what this means, you should probably review a little bit of chemistry to understand a little bit about acidity. But you don't really need to if you understand the basics of pH. Okay, 7-Up is just a way that people use to memorize the sperm pathway. So S stands for semin. Why does it look like this? So looks like a typo to me. I think it's seminal inferius tubules, site of spermatogenesis. Spermatogenesis is basically the creation of sperm cells. That's where it happens, nourished by Sertoli cells, so you should know that. Epididymis, by the way, you have to know these in this exact sequence. That's why people use the mnemonic 7-Up. It helps them memorize the sequence. So you have to memorize the sequence exactly. Semi-inferius tubules to epididymis to vas deferens to ejaculatory duct to urethra to the penis. You have to memorize this exact sequence, and also some of these functions. So the epididymis stores sperm and sperm gain motility. Motility is like mobility, kind of. Vas deferens, raise and lower testes. Okay, now we get into the female reproductive system. So for the female reproductive system, they have ovaries, which have follicles that produce ova. Follicles are like, let me go into follicles a little bit more. Follicles, ovaries. Because follicles are very important to learn because they're part of this whole menstrual cycle, which you also have to learn. Yeah, okay, well, I guess I have to do a whole lesson on the menstrual cycle in a second once I go over follicles. Yeah, so the follicles are in here. All right, and I'll explain more in a second. So ovaries have follicles that produce ova, and they're controlled by these hormones called FSH and LH. And I'll explain further as I do a mini lesson on the menstrual cycle and how the menstrual cycle works. The oogenesis is when you have production of female gametes. So this is in contrast with what we just learned earlier. Where was it? To spermatogenesis. Where is it? Here. To spermatogenesis. So oogenesis is the production of female gametes, whereas spermatogenesis, it doesn't say here, but you should know the definition of spermatogenesis, which is the production of male gametes. Now we go into estrogen, which is the response to FSH, so another hormone. Develops rep tract, thickens uterine wall, Progesterone responds to LH, maintains and protects endometrium. So some people like to memorize it in this way. So estrogen establishes. In other words, it's developing and thickening your uterine wall. So it's protecting, right? Oh, sorry. So it's establishing. My bad. And then some people like to memorize that progesterone protects the endometrium by maintaining it. That's how some people like to memorize it. And you also have to memorize the pathway. Similarly, like you have to memorize the male reproductive pathway, which is the egg goes into the peritoneal sac to the fallopian tube, also slash oviduct. I've never really heard of oviduct that much, but yeah, that's another term. I think you should learn further than that. I think you need to go to like uterus and further than that. Um, Fallopian tube to uterus. Yeah, I think something further a little bit would be better if you knew that too. Okay, let's go into the menstrual cycle and also GnRH. I will review GnRH as well just now, and then basically when I start to teach the menstrual cycle really quickly in the next 10 minutes or so, in the next few minutes or so, it'll be really clear about the hormones that I just mentioned. So estrogen, progesterone, FSH, LH, it'll become a lot more clearer in a second. So the GnRH is another hormone as well, and so FSH is the follicle stimulating hormone, and in males it triggers spermatogenesis, and for females it stimulates the development of ovarian follicles. And then the LH, the luteinizing hormone, in males it causes interstitial cells to make testosterone. So you should know what these cells are, interstitial, and you should know what testosterone is, by the way. And females, it induces ovulation. So I'm gonna go over some of the male stuff first, and then all the female stuff after. So what is a Sertoli cell, first of all? Sertoli cell is a vital supporting cell in the testes, known as a nurse cell, and it nourishes and protects and guides sperm cells. So stimulates that and spermatogenesis. That's what FSH will do. For LH, luteinizing hormone. By the way, I'm pretty sure GnRH is a totally different hormone than FSH and LH. I don't know why GnRH. Let me just make sure. Is GnRH a totally different hormone from FSH and LH? Yeah, so you need to be careful when you use this review sheet by Meltdown because, yes, GnRH is a totally different hormone from FSH and LH. GnRH is releasing hormone made in the brain. FH and LH are trigger hormones made in the pituitary gland. So gonadotrophin releasing hormone is made in your brain, okay? And it's the boss. It sends chemical signals to your pituitary gland to make these hormones. So I'm not saying it's wrong, it just looks misleading to put GnRH, FSH, LH. So this GnRH is made in your brain. In other words, more specifically, your hypothalamus. And your hypothalamus releases that GnRH and it sends it to your pituitary gland. Your pituitary gland will then make FSH and LH. And you're thinking, well, if none of this is on here, so it's not high yield and it's out of scope. No, you have to know this as well. You should know this. Because if you know this, you'll understand the menstrual cycle. All right, so, yeah. And then we'll go over ovarian follicles when I go over the menstrual cycle. So as I said, I'll do the females after. LH, interstitial cells for males. Interstitial cells for males. So interstitial cells are also known as Leydig cells. You should also know the second name, by the way. I saw on one of my Anki cards, and it will probably be useful in the end. Sit in the connective tissue spaces between the sperm, producing semi-infrared tubules inside the testes. Their main job is to make and release testosterone. So just remember that interstitial cells make testosterone. Okay? And now we can finally go over the female menstrual cycle. So I'll set up the lesson. So I remember learning the female menstrual cycle this way. And I learned it from this Redditor's notes, actually. These are not my notes, by the way, but this is how I learned it. So basically, our common knowledge, everyone's common knowledge of the menstrual cycle is like, oh, females have to bleed every month. And that's basically true. Well, they don't have to, but it's part of female reproductive biology. Normal female reproductive biology. So basically, that's our common understanding of it. But here is actually what we define as what's happening, what hormones are being activated, what stages there are, right? So wait 1 second, I'm going to get the graph view first. All right, so this graph view first, I'm going to show as a better overview. So in your overall menstrual cycle, you have a follicular phase and you have your luteal phase. Okay, so okay, we got that. Your days of the menstrual cycle last around 0 to 28 days, and that's just a typical number. Okay, that's why we say every month there happens to be bleeding. But if you're pregnant per se, or the sperm gets the fertilized egg, then you wouldn't really see bleeding, right? But I'll explain that and the reason why in a second. So in the menstruation, what happens is that you're at an all-time low of all your hormones. You're at an all-time low until your estrogen goes up and up and up and up, and then your LH spikes. This is a big key point. Your LH is spiking, and your follicle bursts. So remember, FSH is this growth uh, follicle stimulating hormone, right? It's literally in the name, and so it's stimulating the follicle to grow, and it's growing so much that it bursts, and in the follicle develops the egg, right? And you basically get this burst, and the egg will go into it'll be released into your fallopian tubes. Now the thing is that after this follicle, what happens is it shrivels up into being what we call a corpus. Let me show you now. We can get this image. So now it forms into what we call a corpus luteum, which is like a dead thing, right? It's like dead skin or something. So that's what happens, and you get this corpus luteum, and then your progesterone at that time is at an all-time high, but then it goes back down because it's gonna repeat all over again with a new one, with a new follicle being developed. Okay, so let's go over it in this way. So let's start with menstruation. So you have no implantation, meaning there is no sperm plus egg. No sperm plus egg. So your corpus luteum loses LH stimulation, so you don't have LH. If you don't have LH, then you lose high levels of estrogen and progesterone. So all your hormones are basically going down. LH is going down, estrogen is going down, progesterone is going down. And now your GnRH blocks the removal, uterine lining starts to shed. And that's what we know as your menstruation. The shedding is your bleeding. Bleeding. This is again, our common knowledge of what we know about the female menstrual cycle. But it's not the whole picture of it. Now you have your follicular phase. So your follicular phase is what we call, there's a positive feedback loop that happens. That's why you get a big spike, because there's so much positive feedback that you basically, it grows, and spikes like that. That's your LH spike. Remember that one from the picture? So there's your LH spike here. So then what happens is your GnRH ends up going up. Because it's saying, we're at an all-time low, and so we need to do something about it. It's not good for us to be shedding like this anymore. We can't shed forever. So your GnRH decides to go up. And your GnRH then, which is in from your hypothalamus, which is from your brain, released from your hypothalamus, which is in your brain, and it talks to your pituitary gland to release FSH and LH. So now your hormones are starting to go up in your follicular phase. All right, so GnRH is going up, FSH is going up, LH is going up. That leads estrogen to go up as well. And this is from your ovarian follicles. Now, this forms a positive feedback loop where it goes back, and you will have a loop where GnRH keeps going up and up. So if you have a high level estrogen, then it's just going to keep feeding, keeps going up. High estrogen leads to high everything else, right? So positive feedback loop. But if you have low levels of estrogen, if it's always low, you're just, this is going to be blocked and this is going to be blocked. So you're never really going to get this LH spike. Is not going to happen. Okay, so let's say you got the scenario where we have the high levels of hormones, right? You're going to get the LH spike. And so the LH spike is going to lead to ovulation. Ovulation is going to release, the ovum is going to release the ovary into the abdominal cavity. So in other words, your follicle has grown, right? That follicle that contained the egg, right, has grown. And now that it's grown, and it's basically bursted, right? And then the egg left. The egg left where? It left, according to this, into your abdominal cavity. I just like to say it left into your fallopian tube, so maybe I can find a picture of that again. So yeah, so from here to your fallopian tube, into uterus area. And so it left, right? So now what's left is a dead, here is something that's dead, like dead, it's dead. That's why I call it corpus luteum. This is called corpus luteum now. So now you hit the luteal phase where your progesterone goes up because you have corpus luteum. Corpus luteum is the reason why your progesterone goes up, basically, okay? So now you have a high level of progesterone. It's going to lower your GnRH and your FSH and your LH, and now you have the low level scenario over on this side. I don't know if that's seeable, but over on this side, now you have your low level scenario where you're no longer making GnRH. Your estrogen is down. You're no longer making FSH. Everything's down. Down, and down lead to down, okay? Everything down leads to down, right? In your follicular. So now it prevents ovulation of multiple eggs. So you can't ovulate twice. Now, it could be the case where a woman has twins. So you can have monozygotic twins or dizygotic twins. But if you have, let's say you have monozygotic twins. So monozygotic twins are identical twins from a single fertilized egg that splits into two embryos, right? But dizygotic twins develop from 2 separate eggs fertilized by 2 different sperm cells. So basically, what happened here is a difference in what happened in the luteal phase, where the luteal phase, when you have dizygotic twins, is that multiple eggs would have been probably released. But in normal, like in more normal circumstances, or like more average circumstances, like the lowered GnRH and FSH and LH will block another release. So like you don't want to think of it as just like one follicle is growing. There are other follicles growing in the ovary, right? And so if we have GnRH, FSH, and so much LH, it's going to grow everything, and all the follicles will burst, too many will burst, and the eggs, there will be too many eggs leaving the ovaries into your fallopian tube to get fertilized. If you have too many there, it's not necessarily socially, it's not a bad thing. You can have triplets, you can have quadruplets, whatever. You can have multiple eggs released and fertilized. That's a thing that can happen. But what we're saying is that your body in luteal phase tries to prevent ovulation of multiple eggs. So you typically see like only a birth or only a pregnancy of one baby, right? Right. Okay. So that was the menstrual cycle. All right, so that concludes chapter 2, and the next time would be for chapter 3 on embryogenesis and development. All right, so starting where we left off. Well, technically you're just watching the same video, but I took a break from recording, so I'm back. And now we're on biology 3, embryogenesis and development. So basically, for this one, it's like an extension of the last one based off of cell reproduction, and now we're on embryogenesis and development. The reason why I'm not in dark mode right now is because the babies look like aliens a little bit. But if you're all okay with that, with the babies looking a little bit like aliens, then I mean, they already do a little bit, so I guess we can just continue like that. So let's do this first part over here about, oh, what's my Windows Defender doing over here? Sorry guys. About fertilization. So I already talked a little bit about fertilization, that the sperm needs to meet the egg to get fertilized, and that occurs in the ampulla of the fallopian tube. Okay, so like I said, the eggs after menstrual cycle are leaving and they go into the fallopian tube. The sperm's acrosomal enzymes penetrate. So you should know what acrosomal enzymes are. You should know the term acrosome reaction. And so they penetrate the corona radiate. I think it's actually radiata or something like that, but something like this, and then zona pellucida. So essentially what it's saying here is that the sperm is able to penetrate. I don't know, there's no diagram here. Okay, give me a sec, I'll get the diagram. All right, so as you can see here, you have your zona pellucida at the very outer layer. It's the very outer layer of the egg, right? And then you have your coronal radiata on the very outside. And then you have your zona pellucida on the inner layer. And the sperm is going to penetrate through. So I'm just gonna copy and paste it onto here. So what it's gonna do is it's gonna penetrate through the egg. So this is mine, and it's gonna go in, right? So that's all I have to say about that one. Now we can go back to the review here. So yeah, penetrates those 2 layers of the egg. So 2 layers of egg. This is a typo here. I think it's not radiate, it's radiata. So if you go to pictures, you'll know that. Acrosomal enzymes inject pronucleus. Okay, that's like a lot of memory stuff, okay? Cortical reaction releases calcium, and which depolarizes the ovum membrane and makes it impenetrable. So I'm gonna copy and paste this over to show you a quick lesson about the cortical reaction. So give me a second how to get out. All right, just for a brief quick lesson. So the cortical reaction releases calcium, which depolarizes the ovum membrane. So depolarization, if you don't know what that means, it's related to so we already learned that cells have membranes, and resting cell potential is at negative 70 millivolts. I'm not gonna go too in depth right now because I expect you already to know a little bit about action potentials. So when a membrane gets depolarized away from its resting state, depolarization on the graph kind of looks like it goes up. And so your millivolts becomes more positive when it's depolarized. That's besides the point. This is basically like an electrical signal. And so this depolarization is basically saying that there's an electrical signal that happened. And it makes the ovum, the egg, now impenetrable. Because why? So they tell you why here, cortical reaction, because there's a granular release to empty their enzymes into the space around the egg. The outer coat, zona pellucida, hardens and changes shape. So what happens is the outside layer that we just saw, the zona pellucida, hardens. So this zona pellucida on the outside hardens. So Why? Because it doesn't want any more sperm. Where is it? It blocks extra sperm, and it stops more than one sperm from fertilizing the egg. So you don't want more than one sperm going in here. So that's why that happens. So you have those two reactions, right? Let's go back to the review sheet now. So you have two layers of the egg to memorize, and then you have two reactions to memorize. You have the acrosome reaction, and then you also have this cortical reaction. Cortical reaction. All right, that's good. Now we have a fertilized egg, essentially, at the end of all of this. So I'm going to erase all of this. This new, I'm using my device with the pen thing. It's a little bit slower than my regular, so if it looks a little laggy, I guess that's why. Anyway, so you have your fertilized egg, fertilized, and the sperm got in. Fertilized. Nice. All right, and then what happens is that fertilized eggs become a ball of solid mass of cells, like this little ball of cells, lots of cells called a morula. You just have to memorize that really, and that the morula is a solid mass of cells. And then it becomes a blastula, which implants in the endometrial lining. So let me go get a diagram of the endometrial lining. Endometrial lining. I don't know why everything is in caps. Okay, so your endometrial lining is here. So this is your endometrium. And remember, the egg got fertilized in the fallopian tube, and now it's gonna be in the endometrium while it's a blastula. So that's where it's gonna implant. And upon implantation, that's basically where you start to get, the kid is growing now. But anyway, fluid filled in the blastocoele. So blastocoele, I think, is just another way of saying the blastocoele is just another way of saying the the blastula, but a specific part. So the blastocoele is a fluid-filled cavity. So now we're looking at something that's hollow, right? And it's hollow with fluid. So I mean, it's not empty. There's fluid. All right. And the compartment where it is filled with fluid is called the blastocoele. But the entire thing is called a blastula, right? And then you have your trophoblast, turns into chorion placenta. So these are parts. So your trophoblast is part of the blastula that eventually becomes chorion/placenta. Your inner mast cells become the organism. So let's go look at that. Trophoblast cells, blastocyst. Right, so these are the inner mast cells that eventually become the organism. The trophoblast becomes the placenta. I like to memorize, and the chorion as well. I like to memorize the placenta as like a feeding tube almost. Not, yeah, chorion. It's a little bit oversimplified to just probably say that, but yeah. Placenta is forming, and this inner mast cells is what it comes to, and this chorion is there. And what it was before was the trophoblast. So you just had to memorize that. Okay, now gastrulation, and searching up diagrams will help you. Actually, why was I searching them up? It's all right here. All right, so yeah, where was your chorion? Here's your placenta. Here is your umbilical cord. Chorion's there and stuff like that. Okay, for gastrulation. So now we're moving. The blastula is turning into something even more mature, and that is called gastrulation. So let's go look into it. All right, so this is a good picture. So we know that this is going to mature more, and now we're going to have basically layers. So your embryo is going to start to form in these layers. We have your trophoblast from before, right? And the inner mast cells, we just went over that. And that's going to have eventually an ectoderm. And then after that, you're going to form an endoderm. And then after that, a mesoderm. So basically, the ectoderm is the closest to the inner mast cells. Well, basically, the inner mast cells become a cavity with fluid, right? So it becomes quote-unquote hollow with fluid. So here's another picture of the 3 layers, but essentially you just have to memorize that, after the blastula, the 3 layers in gastrulation will form the ectoderm, mesoderm, and endoderm. Then the rest is memory. So this nervous system forms in the ectoderm, and it's also known as the atractoderm because your skin, hair, and nails, things that people are attracted to, are also developed there, as well as your mouth and the anus. And then for mesoderm, musculoskeleton, circulatory system, gonads, adrenal-cortex, if it helps you memorize it, it's called the muvoderm, right? You can remember meso equals muvo, or even muscle, involved in moving things such as muscles, RBCs. So RBCs are red blood cells and steroids. And then endoderm, you can remember for endocrine glands, but it also involves things that are within. Endoderm is how people sometimes memorize it. So things that are inside, so your GI tract, your respiratory tract, bronchi, bladder, and stomach. So you can think of GI tract like digestive system, like respiratory tract, but not circulatory. Remember, don't get circulatory and respiratory confused there. But most of this is memory. You're gonna memorize that. Memorize this, right? Okay. Actually, I should have discussed this a little bit. Twins. So we already had a discussion about twins and how that happens. So I'm just going to go over briefly that when you have twins, they can be dizygotic or monozygotic. And so our common way of saying it is fraternal, where, you ever see fraternal twins, they kind of look the same, but not really. They look more like siblings. And identical twins, they look identical. And we can say they're monozygotic, meaning they're sharing one zygote. So mono means one, right? And then di means two. So there's two fertilized eggs in the fraternal, whereas there's one fertilized egg in the identical. All right. Awesome. So now for cell. Oh yeah, forgot. I forgot to go over neurulation. Okay, so you have to memorize this order of how the egg becomes this full person, right? Eventually. So basically, yeah, the egg is fertilized, then it comes to morula, then it becomes a blastula, then you have gastrulation, and then you have neurulation. Neurulation is when the mesoderm develops a notochord, and the notochord induces an ectoderm. So remember, this is the ectoderm, and that's developing, again, your nervous system. So this is what process of neurulation refers to, is the development of someone's a nervous system. And remember that in psychology you have, or when you study psychology or wherever, physiology, you have your central nervous system and peripheral nervous system. Now, where do these form exactly? Sorry, this computer is a little bit slower. Where do these form exactly? They form from the ectoderm, and the ectoderm has neural folds, and the neural folds then become the neural crest cells, and the neural crest cells then become peripheral nervous system. In terms of developing the CNS, the central nervous system, the ectoderm has neural folds at some point, and the neural tube becomes the central nervous system. So if you've never seen the neural tube before, it looks interesting. Neural tube. So this is a neural tube, right? And then there's actually different parts afterward that it splits into, like procephalon, mesocephalon, rhomcephalon. And then here's that neural plate here, and that neural plate border, and then they fold to make a neural fold, and that's a neural groove, right? And then, you get your neural tube from there. So this is trying to show neurulation, basically. Okay, so yeah, now we can go into also cell specialization. I think this should have been, I should have talked about it right after doing cell reproduction, cellular reproduction. But cells like to specialize. So all our cell DNA, so I like to, just give me 1 second, I'll get a whiteboard. So I drew this very basic diagram here. And so it just looks funny. So this is you and your body, right? You have all these cells that make up your body and different cell types. So have you ever thought that you have muscle cells, and those are different from your, red blood cells are very different from muscle cells. Maybe like skin cells or whatever you want. All these different type of cells that make up your body. And so, you even have like immune system cells and stuff like that, right? And it's like, where did that come from? Well, it comes from, yes, your DNA leads to you having the cell reproduce. Remember cellular, like mitosis? We had to kind of divide the DNA. So yes, DNA does tell you what cell, but it's not saying, the DNA is not saying specifically, I am a set of muscle cell instructions. It's actually, right, that's just an example of one type of cell, right? But it's actually more like, I have this chunk of DNA here, and this is all the same for all cells. All cells have this chunk of DNA, that makes up you. But what happens is that not all of this DNA is presented, right? Not all of it is selected in a way. So there's like sort of a selection of which DNA. And so we select, this part and this part and this part. And based on this, of our whole image of what makes our DNA, makes up, what makes up our red blood cells, for example. So we get cellular differentiation, in other words. So when we go back to talk about cell specialization, I promise it's related. You get cellular determination, where the cell commits to becoming a certain type of cell. And you also have cellular differentiation, which it follows its determination. So it selectively transcribes genes, right? Selectively. That's what I was talking about earlier, appropriate for the cell's specific function. So if you're functioning as a muscle cell, right, it will select out of the gene. We're selecting maybe only here, here, and here, because we know we want to have a muscle cell. So only this part, these little sections of your DNA are going to be selected. So all the cells, right, have the same DNA. It's just, I have this clump of DNA, but let's say you're a muscle cell, maybe I'll only choose this area. to select for. Or like I have another clump and I want to make a red blood cell instead, so maybe I select only this area of DNA to be so anyway, that's kind of basics of cell specialization. Okay, now we also have induction, where it's just where a group of cells influence the fate of nearby cells mediated by inducers, which are commonly growth factors. So give me a second. So for example, you have something that sends something to the cell and send something to something else. Oh, why is it not working? Send something to something else, right? Sending, and you have this, and it's capturing it. And so a group of cells influence the fate of nearby cells. Sorry, this is not a good idea. This is not a good example. Okay, so there's not many good images on Google. So I would just say memorize it this way. The group of cells, like let's say they're all nearby each other, will influence each other. So that's how you kind of know, like, I'm going to be this way. I'm going to be this certain type of way as a cell, because, you know, it's like you are who you hang around type of quote, right? So that's kind of like induction, but for cells. They are who they hang around. All right, stem cells are this is part of learning about cell specialization. So when your cell first starts out, it doesn't know who it is. You know, like kids, they don't really know what hobbies they like, what blah blah blah, what, I don't know, career, what in their like other stuff. So it's kind of like a bad analogy, but what I'm trying to say is that the cell doesn't know what it is yet. So a totipotent stem cell is when it can be any type of cell. So it doesn't know, right? Pluripotent is when it can be any cell except those found in the placenta structures, okay? And then multipotent cells is when you get a more specialized and can be more different types of cells. So let's look at an example of a red blood cell specialization. Red blood cell specialization. Here we go. So let's say you have oh, well, those are different types of red blood cells. That's not the best image. Let me see. Totipotent. So yeah, their example is one that is here. You have your totipotent cell, which is your morula. It's not really knowing what it's gonna be yet, and then you have your blastocyst, and then your inner mast cells. All right. So that's an example. Pluripotency can also be like this. So you have a fertilized egg, right? And then it's pluripotent after that, because pluripotent is basically where you can be any cell except for the ones in the placental structure. And then you can be multipotent. So you can eventually become, you know, mesenchymal stem cells, neural stem cells. So you can guess maybe neural means I'm going to become, you know, nervous tissue or respiratory tissue or cardiac tissues with multipotent. This is a pretty good image here. Okay, and now we're going to go into cell signaling, because that's actually, you know what, that's kind of related to induction, where cells are kind of like talking to each other, right? So autocrine means the cell talks to itself or signals itself. Paracrine signals is when it signals its neighbor. Juxtacrine signaling is when it kind of uses a neighbor signaling, but it's through a receptor kind of here. And then endocrine signaling, you can't see it well on here, but endocrine signaling means you're going through a bloodstream. So you can think of endocrine as like hormones. They go through your bloodstream, and the endocrine system has to deal with that, right? So I'll just show you juxtacrine signaling. So a cell-to-cell communication, but it requires a direct contact, right? Here's another picture for direct signaling, paracrine signaling, and endocrine signaling. So here's your endocrine with the blood signaling. So yeah, cells have to talk to each other in some way, or else, you know, you wouldn't be able to deliver information like, I don't know, neurotransmitters, you know. So cells need to be able to talk to each other. Okay, kind of communicate with each other, but they're not literally talking, by the way, okay? So fetal circulation, fetal hemoglobin, HbF, is when oxygen increases affinity to HbA. So basically what we're learning here is that fetal hemoglobin is different from adult hemoglobin. So F meaning fetus, and then A meaning adult over here, HbA. So adult, right? So why does the fetus have higher oxygen affinity than adults? Well, it's because, or when we're talking about the adult, we're talking about the mother's affinity to oxygen. And so the baby binds more oxygen. Their HB stands for their hemoglobin, which hemoglobin is part of your red blood cells, and your red blood cells carry oxygen. If you ever learned a little bit about the circulatory system. So then what happens? So basically, as a summary, is that the fetus likes to steal the mother's oxygen, all right? And so O2 and C2 exchange via diffusion. So this is basically, you learned this in the respiratory system. Where did it go? Is there a respiratory system somewhere here? Why is there no respiratory? Did I miss it? I think I missed it. Whoops. Well, let's go through. Let me just get a picture. Yeah, so I literally passed it actually. So this is biology 6 on the respiratory system, and I'll quickly show you how it's related. So basically you have alveoli. There's not even a picture here. Well, so here's a good picture, and here's the structure you'll find inside of your lungs. And so you have these bronchioles and capillaries and alveoli. So there's structures, right? And at your alveoli is where diffusion occurs. Where diffusion occurs, and it's talking about that diffusion of why is it not working well? Of O2 and CO2, right? So if we go back up to the baby over here, that's what's happening as well. So another thing to note about that's interesting about the fetus is that when it's going to the placenta and the umbilical artery, it's holding deoxygenated blood. So that's why it's minus O2 here. That's what it says. Oh, what happened to my... Oh, something happened, guys, to my connection. I guess something happened. I had to pause it. Uh-oh. Let me pause it. All right, so the power went out, so I couldn't really go to image for fetal shunts. I will in a second. Basically, fetal shunts are pathways for the fetus's respiratory, so their breathing system, right? And so it will skip the lungs, and the foramen ovale goes right from right atrium to left atrium. Ductus arteriosus goes from pulmonary artery to aorta, and then fetal shunts will also be able to skip the liver and go from umbilical vein to inferior vena cava, so vena cava or cava, whatever you want to say. So I'm just going to wait till my power comes back on, and then we'll show you the fetal shunts. And then move on to the nervous system, which is awesome because we could finally talk about action potentials, which was related to what we were talking about. Where was action potentials? About here, the cortical reaction releasing, whoops, why is it? Sorry, my computer on this one's slower. Cortical reaction releases calcium, depolarizing an oval membrane and makes it impenetrable. So depolarization and the cellular membrane has to do with action potentials, which I'll explain later after I show you a picture of fetal shunts. Alright, so I don't have my Wi-Fi back, but I'm using my data to just record everything. So fetal shunts are like the pathways, right? But they're different from... The reason why we have to learn them is because they're different from adults and how everything works. So, for example, the foramen ovale, foramen ovale, whatever. I don't know how to say it really, but you can see that it's from over here. It's this little hole, like it looks like a little hole, and so the air can go through. I mean air, but like oxygen, carbon dioxide, whatever. So it's going through here. Let's just say air for now, but I'll specify later. And then ductus arteriosus is up here, right? It's like a hole. So give me 1 second. So I wanted to clarify that basically we're going from the right atrium to left atrium, or the left atrium. The right atrium and left atrium can connect because of this foramen ovale. That's what it's saying here, right? And same thing with the other ones. So because of the ductus arteriosus, the pulmonary artery and the aorta. So the aorta is this big one over here. Aorta and this pulmonary vein here could connect. So before you learn fetal shunts, I would advise to learn about the heart anatomy, which is on cardiovascular system, or learn those together. I know it's a little bit separated. Some things are separated like that, but you can always, go back and see how things connect later on. Okay, so now we can go into the nervous system, one of my favorite parts. So the nervous system is made up of your neurons. So neurons are types of cells, and they are... these types of cells that run your nervous system are called neurons. So nervous system, right? That's the name of this chapter, nervous system. Okay, and so neurons have different types. So there's afferent interneurons and efferent neurons. So afferent neurons will ascend. So the types are based off of like where they are, what they're doing here. So the afferent ones ascend from spinal cord, interneurons are between other neurons, and efferent neurons are exiting spinal cord. So I didn't really learn it this way. I'll show a picture so it's easier and better to do. So I'm going to show you a quick overview picture of the peripheral nervous system. So remember, you have a central nervous system, which is your brain and spinal cord, and then your peripheral nervous system, which is everything else, right? And so then your afferent nerve, it's here. Okay, and then in your spinal cord, you might have an interneuron, and then your efferent nerve goes out. This one doesn't show interneurons, does it? Yeah. Yeah, so here's one where it shows the interneuron. So here's your spinal cord, and then from your hand sensation, you have your afferent, and then efferent as in exiting the spinal cord. So E for exiting spinal cord. So that's one way you can memorize that, right? So A saying like you're going to the spinal cord and like ascending towards spinal cord, towards, and then E for exiting the signal. And so you're thinking to yourself, well, what type of signal is that? Well, if you haven't taken any class before, then this will help give you an overview that it's sending electrical signals. I don't know why it's so laggy. It's probably the computer I have here. Electrical signals. So how it works is that this is your neuron, and these are also your neuron things you have to memorize. So neurons have dendrites where it takes in incoming signals. I think you need to know the function too, which is not written anywhere here, so I'll just go over it, I guess. Yeah, I guess I have to go over that. And then the nucleus and the cell body is also called the soma. Your axon is important. It's like the tail that goes from here to here. And then myelin sheath, faster, means it makes the electrical signal go faster. Okay, if the cell has myelin sheath, if the neuron has myelin sheath, it's gonna make it conduct faster. This conduction happens in like jumps, it makes the electrical signal go faster. Okay, and then nodes of Ranvier, saltatory conduction, that's the conduction that allows the fast speed. So saltatory, it's called saltatory conduction. So we have myelin sheath, right? And basically it jumps. The signal can jump because there's myelin sheath, and it jumps across at the nodes of Ranvier, the electrical signal. That's what it's doing. So even here, this is a picture. So actually, this picture is one of the better ones. So you have your nodes of Ranvier and then your myelin sheath, and the signal is jumping. It's jumping. Here's another good one. So it's jumping. Okay, so it makes the electrical signal go faster. Schwann cells are in your. So these are different types of cells that can make the myelin sheath. So Schwann cells can make myelin sheath for peripheral nervous system and oligodendrocyte for central nervous system. Let me confirm that too. So yeah, they do make myelin. Schwann cells are specialized support cells in the peripheral nervous system, and they wrap around the nerve fibers axons to build fatty covering called myelin sheath, right? So Schwann cells do that for the peripheral nervous system, and the ones that do that for the CNS are called oligodendrocyte, all right? And then you also have the nerve terminal, where this is basically, the name suggests, it's the end of the neuron, and so that's where it's going to connect with another neuron, probably, maybe, and then outward. Or it can connect not to another neuron, but what we call an effector. So I'm going to show a pathway of overview of a connection. So here's an example where you have a neuron going to the spinal cord, sensory neuron, right? Another way of saying like the afferent neuron essentially, and then it's going to a motor neuron, which is the efferent, exiting, right? This is also called a motor neuron because it's going to then allow the special thing to happen where it's going to depolarize muscle cell membranes in order to create the muscle to move, essentially, muscle cells, the muscle to contract, right? So the effector is now, it can be a neuron connected to an effector, is what I'm saying, or it can be like a neuron sending a signal to another neuron, right? So that's what I want to say about nerve terminals, is that it's not just this ending here where nothing else is happening and the electricity just goes into air. No, it's connected to something else to then send that signal over. Okay, summations. So summations are easier to understand once you understand the basics of action potential. So I'll go over action potential first. I actually think glial cells are more important to do first, just because we already went over oligodendrocytes and we just went over Schwann cells right here. So remember, Schwann cells makes myelin for PNS, oligodendrocytes makes myelin for CNS. Okay, so we already went over those, so check mark. And then you have other cells in your nervous system, glial cells, and so you have astrocytes, which are blood-brain barrier, control solutes moving from bloodstreams to nervous tissue. You have epidermal cells. So this is just pure straight memory at this point, which is the barrier between cerebrospinal fluid, so the fluid in your spine. Sorry, not in your spine. You also have cerebrospinal fluid, not just in your spine, but yeah, cerebrospinal fluid and interstitial fluid of the CNS. So the reason I had to correct myself is that you don't just find cerebrospinal fluid in your spine. I remember because I was learning about ventricles back in like university, right? So I remember that the ventricles also hold cerebrospinal fluid, and your brain has basically ventricles are areas of that area, spaces that could have that fluid, you know. So that's there. All right, so that's that. So check mark for that one. And then lastly here, microglia is to digest waste in the central nervous system. All right, so those are other cells in the nervous system, and they're kind of like support cells, and you can think of your neurons as the main cells sending electrical signals. Okay, that's too much annotation that I made there. Okay, now we can go on to, I would like to go into synapse, but it might be too hard to understand. I'm going to actually go through synapse first, because like I talked about beforehand, that your cell connects, your neuron will connect to possibly another neuron, and so that's where we get the synapse. So this is a neuron at its axon terminal. Here's their little leg. This is just like one of their legs, right? At the end of the neuron. We're looking at the end of the neuron here. And so it has these vesicles, like bubbles of stuff, right? Of chemicals. And so neurotransmitters are basically chemicals. And it's trying to send these chemicals over, And the other one, and your dendrite, the other neuron that's receiving, right? Dendrites are for receiving signals, like I said. Are they receiving these neurotransmitters here? And it looks like to be the case that they're receiving also calcium is entering, by the way, calcium. And I'll explain calcium when we talk about depolarization, but this is the connection I was talking about earlier, that neurons can connect with other neurons and also talk to them in a sense. Not literally, but yeah. So then we can go into action potentials, which is more like specifically what's going on behind the scenes. So action potential happens, this all or nothing firing happens when you're, I have to come back to explain action potential. I have to take a break. Okay, so as you can see, these are the action potentials, and then here's the resting membrane at negative 70 millivolts. So here's the resting state, and it's saying that it's at, right, what I just said, negative 70 millivolts. Now, once you hit this threshold activation right here, negative 55, is when, right here, is when the all or nothing initiation of the action potential fires, and this is what we know as depolarization. So you might be wondering what this looks like, kind of like, what does this look like from an overview point? So you have the cell, right? And every cell has this membrane on the outside. And when it becomes, there's channels that allow sodium to come in, or sodium can just come in. And so sodium will come in especially high during depolarization, so high that it hits a peak, and that peak is at plus 40 millivolts. When I studied this physiology, we had to memorize the exact one, so I would recommend memorizing what is resting, what is the resting membrane potential, what is the threshold of activation over here, and then what is the peak of the action potential. And so this, basically, we're mapping out like what's going on in the cell. So at this point, it's depolarization, where lots of sodium is coming in, and so it's making this membrane more and more depolarized. And then what happens is repolarization happens, so you get potassium coming out at this time, and then there's a refractory period, and then it goes back into resting state. So one thing that wasn't on here, I think, was hyperpolarization. I'm going to try to show you this one. Okay, so here you go. That's why I was a bit confused why it was saying this, but you do have a refractory period, yes, but this certain area where it undershoots, my bad, the resting potential, is called hyperpolarization. And for the refractory period, so this is what's happening in the cell literally, right? So resting potential is here, and so this is the sodium and the potassium. And so sodium is gonna end up going in, and then potassium at repolarization goes out, and then hyperpolarization happens where it undershoots, the resting membrane potential, and then the cell goes back. So that's just the basics of the action potential, but I recommend like going a little bit deeper afterward. So then the next thing to know is about the brain over here. So like here's so the brain is made out of white matter and gray matter, right? And so if you look at the brain, white matter versus gray matter. Okay, so here's the white matter and gray matter. So you can see right here, the white matter is actually and your spinal cord is the inverse, where your white matter and gray matter, so your gray matter is on the inside, whereas your white matter is on the outside. And what it represents is your soma, or your cell body, is the white matter, your gray matter is mostly neuronal cell bodies, whereas your white matter is your heavily myelinated axon. So remember we talked about the myelinated sheaths here, the myelin sheath that creates the saltatory conduction, or makes the electrical signal go faster, right? The conduction provides that. So the myelin sheath is creating this white matter part of the brain, and then any gray matter, it could be part of the brain or the spinal cord, and any gray matter is unmyelinated, right? And that means you're also looking at the cell body, so the soma here, right? And then so your brain has white matter deep in and gray matter outer, so you could look at a picture and that'll help you much better. Like this picture is better. So your gray matter is on the outside and your white matter is on the inside for the brain. Went over that. And then your spinal cord, gray is deep, so your gray matter is on the inside, deep within, and then the white matter is on the outside or the perimeter. So that's it for that one. I already explained how the synapse worked. Here, neurotransmitter removed. So I didn't explain this yet, but we'll go onto that last probably. And then after is reflex arc. So sometimes you notice you have a reflex arc, right? So your body can automatically react to things without almost non-voluntarily, right? Non-voluntarily. And so monosynaptic is when you get sensory neuron going to your motor neuron. So right away, like it synapse right to your motor neuron. And this is, and then it goes to your effector, which is your muscle, which is something they didn't write there. So let's just say this is your muscle. And then your polysynaptic is when you get sensory, and then your interneuron and your motor, and then your motor neuron. So motor neurons, what are they? They're neurons that are like basically connected to a muscle. They're gonna be synapsing at a motor junction where they meet with the muscle, basically. So yeah, I already went over central nervous system, which is your brain and spinal cord, and then you have your peripheral nervous system. And so the somatic part is voluntary, and your sensory is afferent, your motor is efferent. So that is good. Voluntary meaning like skeletal muscle, for example. And autonomic is, it could be muscle, right? But let's say you have muscles in your digestive system that you're not necessarily in control of. That would be your autonomic area. And anything that you're not really voluntarily in control of is your autonomic nervous system. And so you have sympathetic and your parasympathetic. Notoriously known, sympathetic is your fight or flight, and your parasympathetic is your rest and digest. So this is what it writes down here. Rest and digest, fight or flight. Sympathetic will relax your bronchi, blood to locomotion, and peristalsis. So peristalsis is basically like it's lowering your digestion movements, right? So you're not resting or digesting, right? So in sympathetic, sorry, you're lowering this. And then for blood to locomotion, the reason why this is increasing, you're getting more blood, is because it's trying to get you for movement. So like it's like you're running from a tiger response, right? So your body thinks you're running from a tiger. It's gonna activate the sympathetic nervous system more so, and so you'll have more blood to locomotion. And then relaxed bronchi, it's trying to be more efficient in cellular respiration and breathing and stuff, because example, your body thinks you're running from a tiger, but anything that makes your sympathetic nervous system fire doesn't necessarily have to be a tiger. It could be exercise, or when your body knows you're exercising or something. And then this parasympathetic system, reduce bronchi, conserve energy, peristalsis. So it's the exact opposite of what the sympathetic system is doing. And then for neurotransmitters, I would better explain this, and also this part. Neurotransmitters are removed from synaptic cleft. So I'm gonna explain. Basically, this area here is your synaptic cleft. Remember I told you that these neurons are kind of like connected in a way. They're not actually physically connected, but they're talking to each other. And so these neurotransmitters are being released from the vesicles, right? From one neurotransmitter, and then it's going to be received on the receptor over here. But the thing is, how are neurotransmitters from the synaptic cleft removed if they're not all received by the receptors? What happens? Do they just stay here out in the open over here? Where do they go? So they can either be broken down by enzymes, reuptake, so it goes back into the neuron that it came from, or it could diffuse out of the cleft. So those are the three ways. And so your neurotransmitters, we were just talking about them, for your preganglionic and postganglionic. So this is basically like saying, say you have one neuron here that attaches to another one here, right? So this would be your dendrites, by the way. Dendrites are here. And so this is your preganglionic over here, and this would be your postganglionic over here. So it's basically saying that your preganglionic neurons are going to release acetylcholine. So maybe I can make a better picture. Just give me a second. All right, so I have the whiteboard open to show you guys. So basically you have your, this is your cell, like this, and then here's your one neuron to another neuron. So basically they're saying that this is your preganglionic, and then this is your post, right? So basically at this cleft is where this one, the preganglionic, is going to release their neurotransmitters. Well, I can't even write nowadays, transmitters. And so if you look at this one here, we're looking at this. So as you could see here, it says neurotransmitters in the sympathetic system will release acetylcholine, and then their postganglionic, epinephrine, and norepinephrine. So postganglionic, epinephrine, and norepinephrine on this side, postganglionic, and neurotransmitter released here would be acetylcholine. And you have to memorize these stuff. I just want to show a better image, so I'm going to find one. Alright, so this is a better, more professional image. So basically you can see here that in the somatic nervous system, there's a central nervous system, and then there's a peripheral nervous system, and then you have the effector organs. So this is your somatic, your voluntary motor system, your voluntary system. So what happens here is that it will release acetylcholine and be able to make the muscle contract this way. So you have this electrical signal running. Remember, the action potential fires, and then the electrical signal runs. And then for the autonomic nervous system, remember we have the sympathetic division and parasympathetic division. So this, all of this really summarizes everything I just went over. So the sympathetic preganglionic will release acetylcholine, and then afterward norepinephrine, and that could lead to any of these effectors over here. So smooth muscle, glands, cardiac muscle, and then it can also be acetylcholine and norepinephrine and epinephrine. And so notice your adrenal medulla. It's not really a postganglionic neuron; it's an adrenal medulla. That's one thing to note. And then parasympathetic division, you have your neuron, and then it sends the signal acetylcholine, and then you get more acetylcholine. So let's see if that one was right. Yep, so you get acetylcholine and then more acetylcholine. So preganglionic right here is acetylcholine, and postganglionic is also acetylcholine in your parasympathetic division. So this is a really good picture that summarizes the neurotransmitter release. Okay, so now we can go over peptide hormones. Most of this is just memory, so yeah, good luck with that. Maybe do either, if you like Anki, do Anki. If you like to do it this way, you can use study tape like that and learn your peptide hormones. So all the peptide hormones are made of amino acids. So if you haven't taken biochem yet, amino acids, I'll just oversimplify it, is like protein. Amino acids make up protein. But yeah, I'll just simplify it for now. But yeah, peptide hormones made of amino acids, cleaved from a larger polypeptide. Okay, then maybe I will have to go over what is amino acid, what is a polypeptide. Golgi modifies and activates the hormone put in vesicles via exocytosis, so I have to go over that as well, like a little bit about cellular transportation. Polar cannot pass through the membrane, so uses extracellular receptors. So I'll go over this peptide hormones in a second. Let's go over steroid hormones. So steroid hormones are made in gonads and adrenal cortex from cholesterol. Don't dissolve and must be carried by proteins. So they must have a carrier with them because they don't dissolve within like the blood to be carried anywhere. They have to be carried by something else, right? They're non-polar, so they can pass through the membrane. They activate nuclear receptors and their direct action on DNA. And examples of steroid hormones include estrogen, testosterone, and cortisol. And then we have some amino acid derivative hormones, which share traits from both peptide and steroid hormones. So an example is catecholamines use GPCR, and thyroxine bind intracellularly. So thyroxine and catecholamines. Okay, so that's a lot of memory type of stuff. I'm gonna go over G-coupled protein receptors. So remember, you have receptors at the ends of these neurons. For G-coupled protein receptors, this is a type of receptor, a G-coupled protein receptor. It's a type, right? It's one of the types. And so notes epinephrine is a ligand first messenger at the end, blah blah blah, phosphorus. So I'll explain this in a second as well, because GPCR is also involved for peptide hormones. So we will do like one lesson of these two together to better understand how they're connected. And then for direct versus tropic hormones, what are they? So direct hormones act directly on the target tissue or organ. An example of that is insulin. Tropic hormones require an intermediary, and they only affect other endocrine tissues. GnRH and LH are both trophic. So remember that in your female reproductive system, there was GnRH. So GnRH and LH are both trophic because remember GnRH was just to increase FSH and LH, right? And so it wasn't acting directly to the effector. It was acting to the pituitary gland, and then the pituitary gland would then release hormones that then act to the effector, right? So remember what the female reproductive system was. And then diabetes. Type 1, no insulin, so glucose is not able to enter cells. Type 2 diabetes, desensitized insulin receptors, glucose unable to enter cells. So I'm going to go over probably diabetes a little bit more too. And then I'm going to go over peptide hormones along with the GCPRs together as like a whole lesson. So one sec. Okay, so here are two pictures I want to show you guys. So you have what we reviewed just earlier, peptide hormones versus steroid hormones, right? And so your steroid hormones, they are made from cholesterol in gonads and adrenal cortex. We know this. We know the synthesis for peptides is that it's a polypeptide cleaved in the Golgi, packaged in vesicles, released via exocytosis. So exocytosis, I'm not going to give a full thing about cellular transport, but exocytosis, you can think of it as exiting the cell, exocytosis via a vesicle. So you're traveling out. It's like astronauts traveling out with a rocket, but these hormones are traveling out via a vesicle, so a bubble they could kind of be inside. So this is like the bubble, and these are like the neurotransmitters in them, right? So they're leaving. So now that so that was exocytosis, a very quick one. And then now hormone travel. So steroid hormones are non-polar, and they're bound to carrier proteins in the bloodstream. So they need proteins. So this is a protein, for example, and this is then your, whoops. How do I get out? Whoops. All right, so it's too laggy. So maybe we need to do the paint tool or something. Protein. So then, let's say you have your bloodstream there. So this is your bloodstream and whatnot. And so your hormones that are steroid hormones can't dissolve, remember? So they need this protein, and they're attached to that protein to help it, like you in a car, you know. But this is more essential. Like this has to happen for it to move around. Like you have to have legs to move around. Okay, I'm just throwing out random analogies at this point. Okay, now we're gonna go into the contrasting side, which is which is peptide hormones. So then they can freely flow by themselves in the bloodstream. They don't need help. They don't need this carrier protein, unlike steroids, right? And so yeah, that's a difference between the two. So peptides also, they are going to be polar, so they're hydrophilic, right? So they will affiliate with water, so that's why they're also okay, like basically dissolving into the bloodstream and being carried out that way. And then they're also binding to extracellular GPCR. So we're gonna go over GPCR, which is the, again, remember the review sheet, the G-coupled protein receptors. Yeah, so peptide or hormones act fast and they have a short-lived response, whereas steroids are slower, right? Okay, so now we're gonna go into GPCRs. I just want to check one more thing. Yeah, so what happens is that to enter the cell, they enter differently. So I want to just go over this mechanism of entering, which is step 3 on the picture. So for step 3, there's different ways that the hormone can enter. So for peptide, it's you have to bind to the receptors, right, to then basically try to get in, so the GCPRs. But for the steroid hormones, they can just go through. They just go through. Remember, our cells have this lipid membrane, so they are whoops, that will allow these steroid hormones to pass through. Okay, so let's go over GCPRs. It's a bit slow, so I'm going to pause and do it on paint. Okay, so here's a simple summary of the 3 steps shown below of how GCPRs work. So in a GCPR, whoa, that is way too big. Step 1 is because peptide hormones are polar, they cannot enter the cell. Instead, they act as a first messenger. So what they're doing when they never really enter the cell, right? So they couple to these protein receptors, as I said before. So these are called extracellular GPCR. Sorry, I've been saying it wrong. It's GPCRs. And this binding changes the receptor shape, activating an attached G protein by swapping GDP for GTP. So if you haven't learned how ATP is made in the mitochondria and stuff like that, or cellular respiration, then GDP and GTP will be kind of confusing. But essentially, if we look at this picture, as you can see, the epinephrine is going to bind to the G protein coupled receptor, right? That's just the step 1 that we just talked about. And then now the G protein is going to, it has a GTP, and it's going to activating an attached G protein. So this is the attached G protein over here, right? It's attached. And then what happens is that it swaps GDP for high energy GTP, which is triphosphate, and this would be diphosphate. In other words, it's getting more energy, is all I'm gonna say. So then in step 3, you get a cascade of the signal where the active G protein activates an enzyme like adenyl cyclase, which floods the cell with many secondary messenger molecules called cyclic AMP. So this part would be over here. Here is the secondary messenger molecules, CAMP, cyclic AMP, and so it's gonna go into protein kinase A. So these secondary messengers amplify the initial signal to create a massive cellular response. So it's like this chain, and then it goes to this one, and then other responses happen in your body, and this is an example of what epinephrine would do. So it would inhibit glycogen synthesis and promote glycogen breakdown. Okay, so endocrine organs and hormones. So for endocrine organs and hormones, you have your hypothalamus. So I've talked about the hypothalamus is part of your brain, and so it can release GnRH, and as we saw in the menstrual cycle, it increases FSH and LH. GHRH increases GH. TRH increases TSH. It's like thyroid releasing hormone, thyroid stimulating hormone, stuff like that, growth hormone. CRH to increases ACTH. So you want to know the actual names, by the way, actual names. That will help on the test. Dopamine, PIF, decreases prolactin, ADH, and oxytocin produced in the hypothalamus, released from posterior pituitary. So one way that will help you also memorize this is like, what effect does this actually have? Like if I increase FSH and LH, like we learned before, what effect does that have on the female system, or even the male reproductive system? What effect do these hormones have? That's another thing you have to note as well, what effect. Right, so then you have your anterior pituitary, and a lot of people use like the mnemonic flat peg. So FSH, LH, acetylcholine, thyroid stimulating hormone. So that's flat, FLH, A-T, and then PEG, prolactin for P, endorphins for E, and GH, growth hormone for G, so PEG. So that's one way that helps people remember that the anterior pituitary releases all of these. And then you have to remember that FSH causes spermatogenesis. Females, it was the growth of ovarian follicles, so we already went over that in the other chapter. Males, testosterone induces ovulation, that's what LH does. ACTH, I think this is synthesize. I don't know about synth. And release glucocorticoids from adrenal cortex. Okay, so maybe it is synth because synth happened twice. And release tridosylribine. Okay, you just need to know that this causes this. All right, you don't have to know that much. This causes this. And thyroxine from thyroid, and this from the thyroid. And then prolactin, increase in milk. For females. Endorphins decreases pain for both male and females. GH increases growth in bone and muscle, and also increase glucose in bone and muscle. And then now you have your posterior pituitary. So your pituitary gland, by the way, just to orient people, pituitary gland is also in your brain, but as you can see, it has two parts. It has an anterior lobe and a posterior lobe. So this is how it looks like. Or you can look at this one, it's better. So there's front side and there's a back side, posterior for back. So ADH is released by posterior pituitary, decreases H2O output in urine and vasoconstriction. Oxytocin increases uterine contractions, milk, and bonding behavior. Positive feedback. So in hormones, as I mentioned during the reproductive stuff, you can have positive feedback and negative feedback loop, right? But oxytocin is positive feedback, and / negative feedback. But here you have positive. Let's go into, let's see, adrenal cortex. Yeah, let's go into the adrenal cortex first. So the adrenal cortex, glucocorticoids, cortisol / cortisone, increase glucose, decrease protein synthesis, and your immune system, sad face. And your mineral corticoids, aldosterone, decrease K plus in the blood, increase Na plus in the blood, increase H2O in the blood due to osmosis, and increase your blood pressure. So one way that can help you guys remember this is think about what scenario would this happen. What scenario would this happen where I have high X hormone, where I have high glucocorticoids, where I have high mineral corticoids? And we'll go over that in a second, but I just want to go through the review sheet first. So androgens converted to testosterone and estrogen in your gonads. Okay, and then let's go into your adrenal medulla, since we did talk about the adrenal medulla, remember? That was releasing epinephrine and norepinephrine. Remember this part? In the parasympathetic and sympathetic, this would be the sympathetic postganglionic epinephrine and norepinephrine release. So remember that picture? Yeah, so your adrenal medulla is releasing those catecholamines, and so your epinephrine is a catecholamine, your norepinephrine as well, and your epinephrine is antihistamine. It increases your heart rate and blood pressure, and norepinephrine increases heart rate and blood pressure. So these are like your adrenaline hormones, fight or flight, remember that, fight or flight, right? And then you have your thyroid gland, and then your thyroid gland, I just remember thyroid for neck because I always remember one of my friends had unfortunately like a growth happen in their thyroid, and they had to get part of it removed before any cancer could happen and stuff like that. So yeah, thyroid gland, just to orient everyone for a picture, your thyroid gland is in your neck. So a good way to memorize stuff again is to relate it back to real life, relate it back to your own life, relate it back to situations that that make sense to you. T4 and T3 are hormones released by the thyroid gland. They're made by follicle cells, and they increase basal metabolic rate. Calcitonin is also released, and it's made by parafollicular cells. It increases calcium in your bone. It decreases calcium in the blood, and it decreases it in the absorption in the gut, but it increases the excretion from the kidneys. And pancreas is related to digestion, so we'll do that in a second. But since we were talking about gonads earlier, gonads release testosterone in testes for male, and estrogen and progesterone in ovaries for female. And then your pineal gland is really cool. I think it's in your brain as well, pineal gland. So yeah, it's in your brain as well. Just to orient people for a picture, it's in your brain. And so it will release melatonin, which controls your circadian rhythm. In other words, your sleep and wake cycle / wake cycle. And then there's the parathyroid glands, which release PTH, parathyroid hormone, which decreases calcium in your blood. It increases decreases calcium in your bone, but increases in your blood, your absorption in the gut. So you can think of the parathyroid gland as a antagonist to calcitonin. So that's another term that's used, is antagonist. So that's another term that's used, antagonist, where it's basically saying like, and you want to know about antagonists as well. So you want to think about, oh, this hormone has this effect. What would be its antagonist or its opposite? It would be PTH, right? So this is its antagonist because it's the opposite. So in the opposite, so where one of them is decreasing calcium in the bone, the other one would increase calcium in the bone. And then pancreas, did we go over that? No. Pancreas, just to orient everyone, pancreas. So I have dust on the keyboard. Okay, so pancreas is near your digestive. Here it's like tucked underneath your stomach almost, right? And so the pancreas will release insulin, glucagon, somatostatin, and as you can imagine, these are all hormones that impact your digestive system in a way. So beta cells are going to be found in the pancreas. You also have alpha and delta islets. So beta and alpha, delta. I want to show if I could a good picture. Yeah, this one's okay, but basically your pancreas has a lot of different cells, and so beta cells for insulin decrease glucose, and alpha cells will increase glucose. So the antagonist to insulin would be glucagon, right? So they're opposites to each other, in other words. And then somatostatin decreases both insulin and glucagon. All right, so that's all the hormones already. So that's great. Now, that was a lot of memory, but if it helps, what you can do is like take a moment to Google a picture of the endocrine organ that you're looking at, and then think about the hormones and try to memorize them. Okay, and also relate the hormones to your real life scenario. For example, when are you in fight or flight? Maybe in exercise, right? You're releasing a lot of catecholamines, your heart rate's going up, your blood pressure is going up, and you're exercising. So think about real life examples like that. Okay, so now we're gonna do review for biology respiratory system. So you need to know about your lungs, and also that it has these walls called visceral pleura. So you have your lung, right? This is inside, right? But your visceral pleura is on the outside. You also have a pleural space. So you have a visceral pleural, which is more of the inside, and then you have a space in between, and then you have another layer, and that's called your parietal pleura. Yeah, maybe light mode makes this look easier to see. Yeah, maybe we'll be in light mode for this one for fun, I guess. So your air pathway is exactly as follows. So it goes from your nostrils, nares of nose is also known as your nostrils, right? Pharynx, which is where food and air travels through. Air is warm, humidified. And then larynx is where air only travels. Your epiglottis covering contains your vocal cords. I'll show a picture later as well. And then you have your trachea, which is ciliated epithelium, collects debris. And then beyond your trachea, there's a bronchi, ciliated epithelium, also debris. And then bronchiole, so it's slowly branching. To the point where you're at your bronchi and bronchioles, you're already in your lungs, by the way. So those are in there. And then alveoli are sacs where diffusion occurs. Surfactant reduces surface tension and prevents collapse of the alveoli. So we're going to go over this in more detail, this prevention of collapse. We're also going to go over quickly a picture of these. So one sec. All right, so this is a good picture. So you have your nostrils or your nares, and then you're breathing it in, you're breathing stuff in, and then it's going down to the pharynx, which is here. Pharynx is this tube here. You can think of them as tubes, by the way. And then your trachea is here. Your trachea is here, and the whole epiglottis thing is this. So your epiglottis helps to make sure that food is going down your digestive system and air is going into your trachea, and no food should be going down into your trachea, into your lungs, right? So the epiglottis is kind of like a gating system. And so yeah, and then it follows the pathway from the trachea into the bronchi to bronchioles, and then alveoli. So maybe I could find a picture. Here's a bronchiole. Is there any better pictures? Tracheobronchial tubes, yeah, I guess bronchioles. And then this is your alveoli, where the diffusion occurs, gas exchange in the alveoli. Okay. All right, so this is the lesson on how your alveoli doesn't collapse and the surfactant thing. So we have a good analogy here about a plastic bag. So to teach how the alveoli stays open, I'm going to use this wet bag analogy. This was made by Gemini AI. So we're going to go through what it's talking about. So the problem is that without surfactant, you can imagine a sandwich bag with wet inside walls. So this is your sandwich bag, and the walls of it are wet. If you press it flat and squeeze the air out, the wet things stick together. So all the air is coming out, and now it's all stuck together. The sides of the bag are collapsed and stuck together. So it's very hard to blow this open again because it's all melted down, right? So you can't, all of this is just a bunch of mush all together, mush. right? And so we can't blow in air again like a balloon and pop it and expand it back to its normal size, right? So then it's very hard to blow it open again. This is what walls does inside your alveoli. The water molecules pull toward each other, trying to collapse. So within your alveoli in your lungs, they have that. And the water tries to stick together, coming inward, trying to collapse. But why does your alveoli not collapse? So the solution with the surfactant is that you have a surfactant in the first place, and you want to imagine the inside of the wet bag with vegetable oil or liquid soap. So your surfactant is like a vegetable oil or liquid soap that can separate this water area, right? And it's like a wall. And then now the walls do not stick together, so the water is not trying to stick together and collapse. The bag inflates easily with a puff of air, so the air is going in easily. Air goes in, and the surfactant acts exactly like that. It acts like a vegetable oil or liquid soap in between, so that the alveoli doesn't collapse. So that is the importance of surfactant. Without surfactant, sad things would happen. You can imagine breathing would be difficult. So yeah. Okay, guys. So now we're on lungs. And so remember contrast this with the fetal system. So let's go back to the fetal system here. So you can see that here the fetus from their umbilical artery out is deoxygenated, but the umbilical vein in is oxygenated. So usually people say that veins are deoxygenated and arteries are oxygenated, but as you see in the fetus, it's not the case. But if you look at your normal person over here, your pulmonary veins are oxygenated and your arteries are deoxygenated, carrying out is deoxygenated to the lungs, and your veins are carrying oxygenated to the heart. I'm going to make sure that's all correct. Right, so your pulmonary vein carries oxygenated blood, right? So, and then your arteries carry deoxygenated. But notice how going back, the umbilical vein is oxygenated and the umbilical artery is deoxygenated. So in an adult, the pulmonary veins carry oxygenated blood from the lungs to the left side of the heart. In a fetus, the pulmonary veins carry partially deoxygenated mixed blood because the non-working lungs do not oxygenate blood. Instead, a small amount of blood flows through the fetal lungs just to nourish tissue, returning low oxygen blood to the left atrium. All right, so now we're going to go to inhalation, so breathing in. So you have negative pressure breathing. So everything about your breathing can be explained by negative pressure, right? And it's an active process that happens. It's not passive. So you have to actively breathe, is what it's saying. And so diaphragm, you have your diaphragm and intercostal muscles, which are your big muscles that help you to breathe. So your diaphragm. So if you ever stretched before or you did yoga and someone says put your hands on your stomach and make a big deep breath, yeah, they were referring to how your diaphragm will move down as you breathe because there's no room, so it's going to move downward. And so what happens is it pushes your organs out where your stomach is, and so your stomach expands with a big breath, like big deep breath. Right? And so increased intrapleural space, increased thoracic cavity, but decreases pressure, increase in lung volume, decrease in lung pressure, and then air rushes in. So here's a better way to look at it. One sec. So I couldn't find too many good pictures, but if you look at this here, you have your atmospheric pressure is equal to your lung interior pressure. When you start to breathe in and you have more space inside of your lungs, the pressure decreases, and so air wants to rush in. And then once the air rushes in, the atmospheric pressure now is less than the lung interior pressure. Your atmospheric pressure is higher than your lung interior pressure. So then air will rush in because there's too much pressure on the outside and the pressure is low on the inside over here. And then what's going to happen is that now atmospheric pressure is going to be too low, and then now your lung pressure is too high because all the air rushed in, right? So now there's so much air in your lungs that you're going to breathe it out. So this is basically what it's saying here, is that when you breathe in, it's increasing intrapleural space, thoracic cavity. Your lung volume is increasing. So your lungs are expanding essentially. And it's decreasing lung pressure. I don't know why it says it twice here, but it says decreased pressure essentially. And then air rushes in. So that's inhalation. And then exhalation is a passive process. As it says here, muscles relax. It lowers your lung volume, obviously, and then it increases pressure. So the opposite of what inhalation does. And then air leaves the lungs. There's also such thing as active exhalation, where you use your muscles to help you force air out. So it is generally passive, but there's an internal there's an active process. Spirometer is to measure your lung capacity, and it cannot measure your total volume. But your total lung capacity, for terminology, is your maximum volume of air that could be in your lungs. Your residual volume is residual air after exhalation. So when you exhale, there's always some air left. And air stays in the lungs to keep your alveoli from collapsing as well. Because remember about the bag analogy, right? About surfactant, right here. Remember that analogy, or go back to it. Vital capacity is the difference between minimum and maximum volume of air in your lungs, and you can see this all calculated here, or mapped out for you here in this visual diagram. Tidal volume is volume inhaled and exhaled in a normal breath, only in your normal breath. This is your tidal volume. right? This is your rest of your reserve for air that's left, the minimum air that's gonna be residuals right here, residual, the minimum that's left over after you've exhaled fully. And then expiratory reserve volume of additional air that can forcibly be exhaled following normal exhalation. So this would be your active exhalation, where you have to actively push to get more air out. And then your inspiratory reserve volume is when you actively push to get more air in above your tidal volume. Your tidal volume, remember, is your normal breath. Okay, so then there's also protection from pathogens. You have viscebrae in your pharynx, mucous membranes, muscular cellular escalator, lysozymes in your nasal/saliva, and they attack gram-positive peptidoglycan. So bacteria, and then mast cells, which have antibiotics on the surface for inflammation and allergic reactions. So mast cells, as you learn later, is part of your immune system, and it's protecting you, right? Bicarbonate buffer, talked about this in chemistry, how it follows Le Chatelier's principle, but basically bicarbonate buffer in biology is basically your buffer system of pH. So it's keeping your pH of your blood the regular pH that it is. So the CO2 plus H2O turns into bicarbonate and then H plus, and yeah. So it's a balancing act, the bicarbonate buffer, to stabilize your blood's pH. Medulla oblongata increases CO2, is hypocarbia, hypocapria, increases respiration, exchange of gases, low O2 is hypoxemia. So I'll explain this with a picture so it's better. So essentially how I like to show it is medulla oblongata. So it's part of your brain. I should see a picture of the brain come up. Yeah, so it's part of your brain stem more specifically, and it's part of the brain that houses your primary respiratory control center, and it constantly monitors other stuff too. But so it has a response to high CO2. So if you have high CO2, there's hypercarbia. That's a condition of high CO2, and so CO2 is the primary driver of ventilation under normal physiological conditions. So excess CO2 diffuses across the blood-brain barrier into cerebrospinal fluid, converting to carbonic acid. So this relates a lot to what we just talked about with the bicarbonate buffer. So when you have abnormally elevated levels, like high levels of CO2, you will have more respiration, meaning you're breathing in more. So you want more O2, right? So this explains the balancing act, right? So if you have high levels of CO2, then you're gonna want high levels of O2 to balance that. Okay, and then if you have hypoxemia, where you have low levels of O2, what do you think is gonna happen for the balancing act? So basically, you're gonna try ventilation and breathe air in and out is what's going to happen. The response to low O2, low partial pressure of oxygen in arterial blood. So when O2 drops significantly, these peripheral receptors that you have send signals to your medulla oblongata, and your medulla triggers an increase in ventilation. So you draw more O2 to your alveoli and raise blood oxygen levels. So raise oxygen because you have low oxygen, right? If you have high CO2, you're going to have higher respiration / ventilation to blow off the excess CO2 is the goal. Yeah, shifting equilibrium in the blood. So these two are very related, so learn them together. That will help you. Okay, so we still have 7, so we'll go through 7 quickly. So this is your blood pathway. So anywhere you see this black arrow, it's deoxygenated, and red is oxygenated. So you have pulmonary, so this is a big overview. Your pulmonary veins bring oxygenated blood to your heart. Your pulmonary arteries blood leaving from your heart carries the deoxygenated blood. All other veins will carry deoxygenated blood to your heart, and all other arteries will do. So notice they're the opposite. So the pulmonary veins are opposite to all the other veins. So all pulmonary veins are oxygenated, but all other veins are deoxygenated. And same thing goes with pulmonary arteries and all other arteries. So they're opposite. So this is the pathway you need to memorize, literally. It might help to see a diagram, but overall, you're going to have to memorize anyway. So just memorize it the way you want to. Your right atrium. So between your right atrium and right ventricle is a valve. It's called the tricuspid valve. So your right atrium to tricuspid valve to right ventricle. Again, black represents deoxygenated blood. Whenever you see a black arrow, and a red arrow represents oxygenated blood. And then from your right ventricle, there's another valve, like a gate almost, right? Pulmonary valve between the right ventricle and pulmonary artery. And to your pulmonary artery goes right to your lungs, to pulmonary veins. Now we're getting back in to the heart, right? So back into the heart is the pulmonary veins, left atrium, the mitral valve is in between, and the aortic valve is in between the left ventricle and aorta, and then aorta to arteries, arterioles, capillaries, right? Capillaries to venules, veins, vena cava to right atrium. So there's definitely better pictures to remember it. Just Google it. We're kind of running over time almost. So electrical conduction. So this is your heart needs electrical signal too. So there's an SA node which pacemakes the heart. It's known as the pacemaker that conducts electricity to the AV node, bundle of His, and Purkinje fibers. I recommend looking at a heart diagram. I can show those later. I'll see how much time is left there. And this is a little mnemonic kind of to help. And then vasculature. Arteries are thick. They are more muscular, elastic, and allows for recoil for the blood to propel forward. Arterioles are smaller muscular arteries, but they're still muscular and thick. Capillaries is one cell thick endothelial wall. So it's kind of thin, really. It's easy to diffuse through oxygen, CO2, and waste. So that's why yeah, we have capillaries and mostly for the diffusion of the gases and even waste. Yeah, thin veins are thin. So opposite to arteries that are thick, veins are thin and they're inelastic, so they're opposite. May stretch to accommodate lots of blood, but they don't have recoil surrounding muscles and they help blood through, and they contain valves. They also have valves. And venules are just small veins. Like arterioles describe small arteries. So yeah. And then we have blood pressure. Blood pressure is systole over diastole. So that's why like 90/60 means 90 systole, 60 diastole. So systole is ventricular contraction, AV valves are closed. Diastole is relaxation, SV closed, blood atria to ventricles. Your normal blood pressure is this, maintained by baroreceptors and chemoreceptors. High blood pressure, increase in atrial natriuretic peptide, low blood pressure. This is what happens in terms of hormones when you have low or high blood pressure. Increased osmolarity increases ADH or vasopressin. And then cardiac output, this is an important formula to remember. It's equal to your heart rate times stroke volume, HR times SV. Okay, and then your blood is considered connective tissue. Connective tissue, this is important to remember because some people don't think it's connective tissue. It doesn't seem like it, but blood is connective tissue. Erythrocytes are the red blood cells, and they're formed in your bone marrow, like your bone, right? No nucleus, they have no mitochondria or organelles. They have no organelles, just remember that. They contain hemoglobin to carry O2. So hemoglobin is the HB we were talking about earlier. Hemoglobin. And then hematocrit is the percent of blood composed of RBCs. So if 80% of my blood was composed of RBCs, that number would be my hematocrit. Leukocytes, also known as white blood cells, are for your immune system. So they're your immune system cells, and they're formed also in your bone marrow. So they're both erythrocytes and leukocytes are formed in your bone marrow. Granulocytes are types of them, and the different types of granulocytes include neutrophils, eosinophils, basophils, and they have control over non-specific immunity, meaning like a general immune response to something like a paper cut, inflammatory reaction. So just like a paper cut, you would have a little bit of a inflammatory reaction, right? A granulocytes include lymphocytes for specific immunity. So a specific immunity could be a specific virus. Monocytes digest foreign matter. If monocytes leave bloodstream for organ, they're called macrophages. That's important. Monocytes, if they leave the bloodstream for an organ, they're called macrophages. Thrombocytes or platelets are cell fragments, and they're for coagulation. So that helps when you have blood clotting and stuff. In a process where blood changes from a liquid to solid gel. So if you ever have an injury, it will help with platelets and special proteins blood called clotting factors can act like a plug so that bacteria doesn't keep coming in, right? So I think of it as like scabbing kind of, or like, blood drying up kind of, I don't know, but not really, I guess. Yeah, so those are also, this is kind of like review for your blood. This is kind of like review and connected really well to your immune system studies in that chapter. I'm not sure which chapter that is. Oh, that's the next chapter. Okay. And then we have blood type. So you can have antigens, which are surface proteins on RBCs, and Rh factor. So blood type is determined by antigens and Rh factor. So whenever someone's saying you're positive or negative blood, they're talking about your Rh factor. So Rh positive is dominant, and an Rh negative person will only create anti-Rh antibodies after exposure to Rh blood. So your body also can be type A, type B blood, and that is given by antigens, which are surface proteins on your RBCs. So you need to memorize this chart, basically. So let's say your blood type A. Yeah, let's say your blood type A, the antigens you produce are A, the antibodies produces anti-B, and you donate to A and AB, and you receive from AO. If you're B, then your antigens produced are B, your anti-A antibodies produced, and you donate to B and AB. And you can receive from B and O. Notice how O is basically this universal donor. If you're AB blood type, you have antigens produce A and B. So this is the antigens you have. Antigens produced, in other words, is the antigens you have, and the antibodies that you produce for AB is none, and then you donate to AB only. You can only donate to AB, but you're a universal recipient. Okay, notice I said O is basically, they don't produce any antigens. So that means they're going to be a universal donor because they have no proteins that are going to be like alarming other people's immune system, right? They're not going to be like, oh, their other people are immune system can't react to nothing because they have none. They don't have proteins that seem harmful in the surface, right, of their red blood cells. So a lot of people, type A, B, and O, like all the blood types, will react normal because there's no surface protein that would seem harmful on the surface. Receive from only O. So O can only receive from O. And then for fluid balance in your body, hydrostatic pressure moves fluid out of the blood vessel into the interstitial fluid around it. Osmotic pressure is the sucking pressure generated by solutes as they draw H2O in the bloodstream. Oxygen is carried by hemoglobin. So hemoglobin, RBCs contain hemoglobin. So your red blood cells contain them. CO2, some is carried by hemoglobin, but most exist in your bloodstream as bicarbonate, and that's why they go over the bicarbonate once again. So I feel like the bicarbonate at this point is so high yield at this point because I've talked about the bicarbonate for 2 times over now on both pages. Coagulation. Coagulation is when the endothelial lining of the blood vessel is damaged and the collagen and tissue factor underlying the endothelial cells are exposed. So the coagulation is related to the thrombocytes that I was talking about over here, this type of immune cell. Coagulation, underlying endothelial cells are exposed. So what happens is your your platelets try to help out, and so prothrombin, fibrogen, thrombin, turning to thrombin, and fibrin clots are breaking down by plasmin. By the way, so basically they try to help like block out this exposed area. So underlying endothelial cells are exposed, and what your body's trying to do as an immune response is try to make sure no bacteria is going to get in. So they're going to try to block that out. Yeah. Okay, so for the immune system here, now we're on 8, is a lot of stuff. It looks intimidating, but so you have a basic structure of your immune system, can be divided into innate immunity and adaptive immunity. So innate immunity, I actually just prefer dark, is where defenses that are always active but non-specific. So if you get a small paper cut or something or whatever, skin, mucus, stomach acid, and tears are part of your innate immunity. They're helping you. Your skin, a lot of people don't think it's part of your innate immunity, but it acts as a really good barrier against, harmful things, right? So the moment you get a paper cut, then you've activated your system, and it's like, oh my goodness, bad stuff is going to come in now because your skin is actually a really good barrier from all this bacteria and stuff you don't want inside your body. And your adaptive immunity are defenses that take time to activate and is specific to an invader. So you can think of a specific virus or something that comes into your body to attack, and that will activate more so of your adaptive immunity. And it's not that one activates one and the other then activates in its own time. Your body doesn't work like one or the other type of thing. But let's go into innate immunity first because, well, that's the first one that's usually introduced in courses. So yeah, its non-cellular innate defenses include your skin, mucus, lysozymes, complement system, and interferons. So your skin is the physical barrier. I just talked about that, and it secretes antimicrobial enzymes like defensins. Mucus is on your mucous membranes. They trap pathogens, and they're in the respiratory system. Mucus is propelled upward by cilia via mucociliary escalator. Lysozymes are in your tears and saliva, and they're antimicrobial compound. Complement system can punch holes in cell walls of bacteria, making them osmotically unstable, leading to lysis. So basically, they can punch holes in bacteria and make bacteria like explode and die. Yeah. And also triggers opsonization. So I'll go over opsonization in a second. Interferons given off by virally infected cells. So interferons interfere with viral replication and dispersion. So they're helping with protecting you. So opsonization is basically, I'm pretty sure, a tagging system. Yeah, so it's a tagging system where opsonization is an immune process where harmful germs or dead cells are coated with proteins called opsonins. And so things in your adaptive immunity can then find opsonins or phagocytes, which are immune cells, can find it and destroy and swallow up those invaders. So phagocytes are basically able to, let's see where they are, phagocytes. I don't see them here. All right, well, yeah, opsonization is explained again here, where antibodies mark pathogens for destruction. So it's basically like a marking system where the body's saying, hey, this is dangerous, right? And then cellular innate defenses include macrophages, MHC1 and MHC2. These are antigen professional. They're going to be presented to professional antigen presenting cells, but we'll go over that in a second. Dendritic cells are also involved, natural killer cells, and granulocytes. So macrophages ingest pathogens and present them on MHC2, and they secrete cytokines. So cytokines are like chemicals that say, hey, this is the area we need help with, guys, and it recruits other immune cells to come. So they are like your first actors of defense. After, like, let's say your skin gets exposed, your macrophages are working inside of you, and they're releasing cytokines. They're trying to kind of invite other cells cells to come over and help. MHC1 is present in all nucleated cells, displays endogenous antigen to cytotoxic CD8 cells. So how you can remember it is that CD8, if you do 8 times 1, you get 8, right? MHC1. This is just a memory tip. And then for MHC2, you have to remember that it's displaying to CD4 T cells, not CD8 T cells. So MHC1 is CD8 T cells. MHC2 displays the antigen, displays the harmful thing, right, to CD4 T cells. So one way to memorize it is 4 times 2 is 8. Right? So both of them lead to 8, but CD4 goes with 2, that makes 8. So 4, CD4 for T cells, 2. So that's one way to memorize it. It's not how you have to, you can just memorize it. And then dendritic cells are antigen-presenting cells in the skin, and natural killer cells are attack low on MHC, including virally infected cells and cancer cells, and granulocytes are little ones important. There's different types. There's neutrophils, eosinophils, and basophils. So neutrophils are activated by bacteria, and they conduct phagocytosis, so they eat stuff up. Eosinophil is activated by parasites and allergens, and they increase histamines. Basophils are activated by allergens, and they inhibit your blood clotting. Okay, so that is basically all of your your innate immune system. I will go over your lymphatic before going over your adaptive, just because I want to show how it connects. So your lymphatic system is like your circulatory system, but for your immune system is how I like to think of it. A circulatory system that consists of one-way vessels with intermittent lymph nodes, provides mounting immune responses, connects to cardiovascular system via thoracic duct in your chest, so it does connect to your cardiovascular system, equalizes fluid distribution, transfers fats and fat-soluble compounds in chylomicrons. Edema results when lymphatic system is overwhelmed and can't drain excess fluid from the tissues. So edema is basically swelling, by the way. Yeah. And a lot of your stuff from your immune system comes from the, let me show this, lymphatic system. Okay, here. So the lymphatic system, as it says here, is a vital part of the immune system. It acts as a transit and filter network. It moves fluid called lymph through your vessels to small organs called lymph nodes. These nodes house white blood cells. Remember, white blood cells are your leukocytes that trap and destroy germs like bacteria and viruses to keep your body safe. And then it goes over main cells in your lymphatic system, and those happen to be the B and T cells. That's part of your adaptive immune system. And then they also have like the macrophages and natural killer cells, which are part of your innate immune system. So it's carrying immune cells, which I don't know why it didn't explicitly say that, but yeah. So it's like a transmission system where they all are going across your body. For adaptive immune system, for humoral immunity, it centers around antibody production by B cells. So it kills antigens, so the bad stuff, while they are floating around in the fluid humor. Fluid is also called humor. And so your B cells, or your B lymphocytes, these are specific types of white blood cells made and mature in the bone marrow. So we already went over that they're made in bone marrow, activated in the spleen or lymph nodes, and they express antibodies on its cell surface. Antibodies are produced by plasma cells, which are activated by B cells. So B cells will activate antibodies, and they can target an antigen, so something bad, and they contain 2 heavy chains and 2 light chains. So what that means is if you go to a picture of an antibody, you'll see this picture. There is 2 light chains and 2 heavy chains. So here are the light chain, and this is the heavy chain. So light chain is smaller. And then hyper and constant region and variable region, tip of the variable region is the anti-binding region. So this is talking about variable region here. Here's your antigen binding site. So that's what it's talking about. Hypermutation, mutation of antigen binding site on antibody results in varying affinities of antibodies for a specific microbe. And there's 5 different types. Okay, and then we already talked about opsonization, so we'll go over to agglutination, where pathogens will clump together into insoluble complexes caused by opsonizing pathogens. So agglutination is the clumping together of things, right? Memory B cells lie in wait for a second exposure to a pathogen. So let's say you went and got your vaccinations done. That would have been your first exposure to, let's say, a specific type of flu that was happening that year. Now, memory B cells, like in the name suggests, they memorize that, oh, that's dangerous, so I know for next time how to protect myself. So we'll have a secondary response, more rapid and vigorous, meaning it knows right away what it's going to do as a defense against when that secondary exposure happens to that virus that came over or some pathogen. Okay, cell-mediated cytotoxic immunity centers on T cells. So we're not centered around B cells like in humoral immunity. We're centered around T cells in cytotoxic cell-mediated immunity. And T cells are even more dangerous because they kill things right away. So they respond to cells once they have been infected by an antigen. T lymphocytes or T cells are made in your bone marrow as well, but they mature in the thymus, and they coordinate immune system and directly kill infected cells. So I was saying they're pretty dangerous. Cell-mediated immunity. So there's positive and negative selection, maturation of T cells, facilitated in your thymosin and occurs in the thymus. Okay, so that's your maturation. Remember I said earlier. And so positive selection is maturing only T cells that can respond to the presentation of an antigen on the MHC. So remember MHC over here. Negative selection is when it causes apoptosis in the T cells that are self-reactive. So if the T cells decide to like destroy its own body, if your T cells are destroying itself, it means there's negative selection, and your body doesn't want your body to destroy itself. So it's gonna apoptosis in T cells. It's gonna basically those T cells are gonna die. Because yeah, that's negative selection. And so helper T cells are, well, in the name suggests, or CD4 cells respond to antigen on MHC2. So we already talked about that over there. Coordinate rest of the immune system, secreting lymphokines to activate immune defense. TH1 secrete interferon gamma. TH2 activate B cells in parasitic infections. So a lot of memory stuff. Cytotoxic T cells, also known as CD8 cells. This is the most high yield. These are other terms that you might see for them. Also known as killer cells, respond to antigen and they just kill the virally infected cells. Suppressor T cells, or Treg, downregulate the immune response after an infection and promote self-tolerance. So at some point, your immune response can't be all that high all the time, right? So your body, because it's using resources, and your body doesn't want to do that all the time. So afterwards, we have T cells that suppress the immune response. And so defective suppressor T cells lead to autoimmune conditions. So if your immune response is so high, and you keep having this immune response, you might have defective suppressor T cells, which are not downregulating your immune response. You're upregulated, where it keeps killing stuff, and it's killing too much stuff. It's killing stuff that is actually needed for your body, which leads to autoimmune conditions. Autoimmune, by definition, is like your body self-attacking itself. Memory T cells serve a similar function to memory B cells. Autoimmune conditions is a self-antigen is recognized as foreign, and the immune system attacks its own normal cells. So when you have an autoimmune condition, it's basically your body attacking itself, and a self-antigen is recognized. So it sees itself as an antigen. It sees itself as dangerous and attacks itself. Allergic reactions or non-threatening exposures incite an inflammatory response. Immunization induces active immunity, activation of B cells that produce antibodies, and passive immunity, transfer of antibodies to an individual. So that would be breast milk. So that would be when you were a baby and you were ingesting breast milk, then that would be an example of passive immunity, transfer of antibodies to that baby or individual. And it can get very dangerous if that's why it can be very dangerous with blood types. If the baby and mother's blood types are incompatible, there can be a lot of problems there. Anyway, digestive system is chapter 9. So yeah, we'll go over that. So digestive system is just an overview. You have your intracellular digestion, extracellular, and also mechanical digestion, and then chemical digestion and peristalsis. So we already talked about peristalsis, which is rhythmic contractions of the gut tube, like movement. So in your sympathetic system, it'll lower. So remember sympathetic fight or flight, so you're not really trying to digest, right? So it's lowering peristalsis, and then increase parasympathetic. It will increase these rhythmic movements of your digestion in your parasympathetic, when your parasympathetic nervous system is more so working. And then intracellular digestion is oxidation of glucose and fatty acids to make energy, but extracellular digestion is the process by which nutrients are obtained by food, occurs in alimentary canal. In other words, intracellular is happening on, like, I guess literally, like cellularly you can think of, but extracellular you can think of like, oh, you're just eating food and it's going through these tubes. And then mechanical digestion is the physical breakdown of the large food, so basically chewing food down. That's a physical mechanical digestion. And then chemical digestion is the, like, what your stomach does. It has enzymes that can cleave the chemical bonds, such as peptide bonds of the proteins or glycosidic bonds of starches. So carbohydrates and proteins that you're eating can be broken down chemically. So that's chemical digestion. That happens like in your stomach. Digestive pathway, you need to just memorize that: oral cavity, the pharynx, esophagus, the stomach, small intestine, large intestine to your rectum. Feeding behavior hormones. So basically, if you have ADH and aldosterone, you have increase in thirst. Increase in hunger is because of glucagon and ghrelin. And then leptin and cholecytokinin increases your satiety, so you get full easily, like you feel full easily, so your leptin and cholecytokinin is high. And then for your duodenum is the first part of your small intestine, a basic pH of 8.5 environment, site of majority of your chemical digestion. So this is actually where most of your chemical digestion happens. Doesn't happen in your stomach, right? Enzymes in duodenum, disaccharides, brush border enzymes that break down maltose, isomaltose, lactose, and sucrose. So these are basically your carbs that are being broken down, carbohydrates that are being broken down into monosaccharides. So they're basically complex carbs, so polysaccharides that are being broken down into monosaccharides. Aminopeptidase and dipeptidase, brush border peptidases. So enzymes, enteropeptidase, another enzyme activates tryptosinoglin, and procarboxypeptidases. So a lot of chemical breakdown happening, and these are the enzymes that are helping with it. Hormones in duodenum include secretin and cholecytokinin. So secretin is your peptide hormone, stimulates release of pancreatic juices and slows motility. Cholecytokinin stimulates bowel release from gallbladder and release of pancreatic juices in satiety. So this is a lot of memory-based stuff. Your oral cavity, you should already know what that is. That's where your mechanical digestion is. You're literally just chewing. Salivary amylase and lipase are start the chemical digestion of food. Bolus is just another term for like food in your mouth that is going to go down and be swallowed. It's basically chewed up food. And then pharynx connects your mouth to the esophagus. The epiglottis prevents food. Remember I said the epiglottis is kind of like a gate. So it doesn't want, also it's a gate that doesn't want any food to go into your lungs. So yeah, that's why it prevents food from entering your larynx and then down your trachea. So it should not go down your trachea, any food. Esophagus propels food into to the stomach using peristalsis. Top third has skeletal muscle and is under somatic control. Bottom third has smooth muscle. Middle third has a combo of both. The middle and both are under autonomic control. So by the way, smooth muscle is under more so autonomic control, and skeletal muscles are, because we haven't done our muscles review yet, it might be confusing, but skeletal muscles are always under like voluntary or somatic control is what they say. And smooth muscle is usually under autonomic control, meaning you're not really telling yourself, you know, you need to contract type of thing. Your stomach has an acidic environment. The pH is 2, so it's very acidic. 4 parts include the fundus, body, antrum, and pylorus. The enzyme pepsin chemically breaks down the proteins. Secretory cells that line the stomach include mucus cells, chief cells, parietal, and G cells. So your mucus produce mucus. These cells produce mucus, which protect stomach wall from the acid or the acidity, right? Chief cells secrete pepsinoglin, a protein activated by the acidic environment. Parietal cells secrete HCl, an intrinsic factor, which is needed for vitamin B12 absorption. G cells secrete gastrin, a peptide hormone that increases HCl. So these two are kind of working together. After processing in the stomach, food particles are now called chyme. And chyme exits through the pyloric sphincter into the duodenum. So knowing what food is called afterward is important too. And then absorption and defecation. So the jejunum and ileum are basically parts of your small intestine, or they're names for parts within your small intestine, and they're primarily involved in absorption. So they're trying to absorb nutrients, right? So the small intestine is lined with villi, which are covered with microvilli. So villi have capillary beds which absorb water-soluble nutrients. Lacteal absorbs fat, absorbs fat and sends that to your lymphatic system. Remember, your lymphatic system is also involved with your immune system. And then now your vitamin absorption. There's fat-soluble vitamins, A, D, E, K, and they enter lacteal. And there's also water-soluble vitamins, which is all others, which enter your plasma directly. For your large intestine, it absorbs H2O and salts and forms feces. The cecum outpocketing that accepts fluid from small intestine through ileocecal valve, site of attachment of appendix. So it would help just to show a picture of this. So whenever you have the time, just type in some pictures for review as well. Structure of colon. So your colon is ascending, transverse, descending, and sigmoid. So also I'll show the cecum, and then also the it's over here. So here's your cecum. And then here's now your colon. You're ascending, transverse, descending, and sigmoid. Okay, and then gut bacteria produce vitamin K and biotin B7. And then you also have accessory organs such as your pancreas, liver, and gallbladder. They originate from your endoderms. Remember we talked about cell replication and reproduction of things. And endoderm is part of one of the layers during gastrulation, I think it was. And so pancreas, acinar cells produce pancreatic juices that contain bicarbonate pancreatic amylase, pancreatic peptidases, and pancreolipase. Liver synthesizes bile, albumin, clotting factors, process nutrients, detox. So detox is turning this ammonia to urea, and so that you could like pee it out basically, as well as alcohol and drugs. So it's detoxing your body. Liver receives blood from abdominal portion of the digestive tract via hepatic portal vein. And then your gallbladder stores and concentrates bile, CCK. That's important. CCK stimulates bile release into the biliary tree, which merges with pancreatic duct. So your excretory urine pathway is related to basically this detox and urea thing over here, where you go to the washroom for what reason? Why do you have bladder that goes to your urethra and out, right? Because basically your body's trying to remove waste, right? So this is the pathway of the urine pathway. It goes from your Bowman space to within your kidney, right? You have this is like a picture of like a nephron, which is a part of the kidney. So functioning unit of the kidney. So everything's a little bit not, it looks organized, but it's not that organized, or how I would teach it. So the nephron is here, and so Bowman space is right here, and the proximal convoluted tube is right here. Your descending loop of Henle is literally descending, and it's going down, and it looks like a loop here. Your ascending is up here, and that's the ascending loop of Henle. Remember, we're looking at a picture of a nephron, right? This is a distal convoluted tube right here, and then this collecting duct is over here. Collecting duct, and then your renal, pelvis, uterine, bladder, and urethra is somewhere else, not in this picture. It's on your body though, and it's in lots of like anatomy diagrams. So then your kidney contains the cortex and medulla and produces urine, which dumps into the ureter and renal pelvis. Urine is then collected in the bladder until it's excreted through the urethra. So yeah, so your kidney is processing all the stuff and it's trying to collect waste essentially, and then get it to your urethra so that you can go to the washroom and excrete all that stuff. And so yeah, the functioning unit of your kidney is the nephron, and so the nephron has that proximal convoluted tube over here, as I showed, which is a site of where you have most of your reabsorption of important stuff like glucose, amino acids, vitamins, salt, H2O, but it's also a site of secretion. And then your descending loop of Henle, which is this one, is permeable to H2O but not salt. So that means the filtrate, which is moves into the more osmotically concentrated renal medulla. So this medulla is on the outside. So because the renal medulla is concentrated, the filtrate wants to move into that concentrated area because there's a lot of water. So water is reabsorbed from the filtrate into your body. Counter current multiplier system. So the vasa recta and nephron flow in opposite directions, creating a counter current multiplier system that allows maximal reabsorption of water. Maximal reabsorption of water. Ascending loop of Henle, we just talked about, is permeable to salt but not to H2O. So that's important. Therefore, the salt is reabsorbed both passively and actively. So the diluting segment is the outer medulla, so the one that has more water now. Because salt is actively reabsorbed in this site, the filtrate becomes hypotonic compared to the blood. So hypotonic meaning the cell swells. Here. The cell swells with a lot of water, H2O coming in. Okay, compared to the blood. And so distal convoluted tubules over here, and so it's responsible to aldosterone, the hormone, and is a site of salt reabsorption. So you have salt reabsorption and waste excretion, like the PCT. So it is like the proximal convoluted tube. So the proximal and distal convoluted tube are kind of like each other. And collecting duct is responsive to both aldosterone and ADH, and has variable permeability, which allows reabsorption of the right amount of H2O depending on the body's needs. So it's permeable depending on what it needs. Remember the proximal tube. The proximal tube is right here, and remember that in your descending it's permeable only to H2O but not salt. So the filtrate moves into more osmotically concentrated renal medulla, so water is reabsorbed from the filtrate, and your ascending loop of Henle is permeable only to salt but not to H2O. Okay, so yeah, the kidney contains a cortex and a medulla and produces urine, which dumps. So we already went through that. Your renal portal system is two capillary beds in series. So we're talking about blood, right? So the blood flow goes from your renal artery to afferent arterioles to glomeruli. So basically it's describing this renal portal system, which is a bunch of veins and stuff happening around this nephron, right? So glomeruli, efferent, vasa recta, which surround the nephron and renal vein, right? So you have veins near your in your kidneys, right? Filtration, Bowman's capsule moves solutes from blood to filtrate. So there is a Bowman's capsule at the very beginning. So this is what they call the Bowman's capsule here. Direction and determined by hydrostatic oncotic pressure differentials between the glomerulus and Bowman's space. Secretion, the movement of solutes from blood to filtrate anywhere other than the Bowman's capsule, and reabsorption is the movement of solutes from filtrate to the blood. Their pH is kidney can regulate pH with bicarbonate and H+. So this is like a bicarbonate buffer. Aldosterone is a steroid hormone synthesized in adrenal cortex in response to angiotensin II or high potassium, and is derived from cholesterol, increases sodium reabsorption in the distal convoluted tube and collecting duct. So it increases H2O reabsorption, and as a result, it increases your blood pressure, but there's no change in blood osmolarity. Vasopressin ADH is a peptide hormone synthesized by the hypothalamus and released by posterior pituitary. It increases permeability of the collecting duct to H2O. So basically, remember that your collecting duct can be responsive to aldosterone and ADH, and ADH is a really good hormone that helps tell it like, hey, we need more or less water. So that's why the collecting duct can be variably permeable, meaning it could be permeable to water or it could be less permeable to water depending on what the body needs, and ADH vasopressin helps it signal to it, hey, we need more, we need less water. Okay, your bladders has a destrucer muscle, internal urethrosphincter, and smooth muscle, external urethrosphincter is the skeletal muscle. Your destrucer muscle is a lining and it's under parasympathetic control, so relaxed. Think of relaxed. Parasympathetic for internal, and then external is a skeletal muscle, so it's always voluntary. So one way to remember skeletal muscles and stuff is skeletal muscle is always voluntary and smooth muscle is always under parasympathetic control or autonomic. In other words, so then, or not in other words, parasympathetic and autonomic are different things, by the way. Skin. So your skin has an epidermis, dermis, and hypodermis. So these are different layers of the skin. For your epidermis, you'll also have layers within the epidermis to memorize too. Stratum basale, stem cells to neurocytes. Stratum spinosum have Langerhans cells. Keratinocytes die on the stratum granulosum. Stratum lucidarum are only on thick hairless skin. Stratum corneum, molten layers, flat keratinocytes. So it's just like mostly a lot of memory here. Langerino cells, macrophages that are antigen presenting cells in the skin. Melanin are produced by melanocytes, protects the skin from DNA damage. So yeah, melanin. Dermis is a papillary layer and reticular layer. Sensory Merkel cells. So these are sensory cells. They can detect things. So Merkel cells detect deep pressure and texture, free nerve endings for pain. Mesenter's corpuscles is light touch. Ruffini endings can sense stretching. Pacinian corpuscles is deep pressure and vibration. The hypodermis, it has fat and connective tissue and connects your skin to the body for thermal regulation. So your skin is part of thermal regulation because you'll notice that you could have sweating, so evaporative cooling. So basically, water is exiting your body, and it's taking heat out of your body through that water. Sorry, not through that water, but like, yeah. And then piloerection warming. Piloerection is another way of saying goosebumps, basically. Let's see, shivering is another way to warm your body because like in the winter you're really cold, for example, that movement of your muscles is creating heat. Vasodilation and vasoconstriction. So if your blood vessels or blood vessels are vasodilating, it's for cooling, and vasoconstriction is for warming. I'm just going to check that real quick. Yeah, so vasodilation makes your skin feel warm, but its actual job is to cool down your body. So vasodilation is to help cool down the body, whereas vasoconstriction is to make your body more warm. So this is ways that your body helps thermal regulate itself in terms of temperature. Okay, we have two more, which is great. We have the muscular system. So it would help to take a physiology class, but you really don't need to. So you have your skeletal muscle. We talked about skeletal muscle many and multiple times. It supports movement, blood propulsion, thermal regulation, and they're striated. And they are under voluntary somatic control, and they're multinucleated cells. So why are they striated? It's because they have two types of fibers. They have thick and thin filaments, thick and thin filaments, and that's why you get striated muscles, which we'll talk about later. Red fibers are slow twitch muscles, and they support they're for support. Active are the white fibers, and they're for fast twitch, so they're for anaerobic metabolism, whereas the red fibers carry out oxidative phosphorylation. For oxidative phosphorylation, this is part of the final stage of aerobic respiration, which means you're doing more so aerobic activity. So an example when you're using mostly red fibers is, for example, a marathon runner. They're using a lot of oxidative phosphorylation, whereas like a fast sprinter is using a lot of their anaerobic metabolism, so they're using a lot of their white fibers. Okay, there's also different types of muscle, right? So skeletal muscle is just one type, and then you also have smooth muscle, which is for respiratory, reproductive, cardiovascular, digestive, and it's involuntary. So your skeletal muscle is voluntary, whereas your smooth muscle is involuntary, or also known as autonomic. It's only uninucleated. It's not multinucleated, like your skeletal, and it can display myogenic activity without neural input. Cardiac muscle is another type of muscle. It's a contractile tissue of the heart. So anytime you hear like cardiac, it's kind of like anything to do with the heart. And so involuntary, it's under involuntary autonomic control. You're not telling your heart when to beat. It's just beating on its own. Uninucleated and sometimes binucleated can display myogenic activity. So myogenic activity, by the way, genic activity is intrinsic the ability of muscle cells, particularly smooth muscle and cardiac, to contract and relax spontaneously without any other stimulus. So that's just what it means about myogenic activity. Cells connected with intercalated discs and contain gap junctions. So this is a key feature of cardiac cells, which the others don't have, is the intercalated discs that have gap junctions. And remember we talked about cells talking to each other. I'm pretty sure gap junctions are an example of juxtacrine communication. So juxtacrine signaling. So yes, gap junctions are a form of juxtacrine signaling where the two cells are connected and they're basically directly talking to each other in a way. So this is a good overview of chemical signaling, but we already went over that in the first parts of the video. So then you also have your skeletal system, so your entire skeletal system. But before we go into that, I'm just going to look over, yeah, this is your bones, right? Okay, before you go into your bones, so when I explain sarcomeres, because they're more in-depth about your how your muscles work. So sarcomeres are the basic contractile unit of striated muscles, so your skeletal muscles. And so how do I get, for example, my bicep to contract? So let's just do the basic description. So your sarcomeres are made up of thick myosin and thin actin filaments. Myosin and actin. Myosin is thick and actin is thin. And because of that, you get striated pattern looking. Let's see if I can get a picture. Striated muscle patterning. Yeah, so the striated patterning of people's muscles is because of the the thin and thick filaments, right? So this is a good picture. This is a good picture of your skeletal muscle. And so let's go back for a second. Troponin and tropomyosin are found on the thin filament, and they regulate actin-myosin interactions. So on your thin filament, you have troponin and tropomyosin. And so you basically want to understand, how does your muscle, how is that able to contract? That's what we're trying to describe right now. So one second, I'm gonna get a picture. So here's a good picture of what's happening. So you have this myosin binding sites covered by tropomyosin. So they're covering these binding sites, right? Tropomyosin is covering binding sites. So tropomyosin is found on the thin filament. So think of the thin filament as like a rope, and the rope has this protein on it, tropomyosin, and it's blocking this actin-myosin interaction. So it's blocking this area where actin can interact with myosin. But once, you'll see later that we are able to remove tropomyosin from covering the area, and so then actin and myosin can then work together, and at that point you can get a muscular contraction. So myosin binding sites are exposed when troponin binds calcium. So give me a sec. All right, so I found a picture here of the sliding filament theory, and you should learn this. It's not directly on there, but it should be explained. So you have this myosin head, right? And so cross bridges are formed between myosin and actin and myofilament. So actin, again, is on this thin filament. Actin is your thin filament, right? And so you have your myosin head, and there's energy here, right? ADP, and there's a phosphate there. So once the cross bridge forms, the myosin performs a power stroke. So it's moving, it's like stroking inward, this way, inward, and it's using energy. So ADP and P are released, so it's using energy to do this power stroke. So it's pulling inward. So this is why, okay, this diagram is good, but like, it might be confusing for some people. But yeah, it's pulling this rope in, you could kind of imagine. And so where was our picture? Okay, here it is. Is this our picture? Yeah. Okay, so new ATP then attaches to the myosin head, and cross bridge then detaches. So when a new ATP attaches to the myosin head, it will detach from this this thin filament here. And then what happens is that, and by the way, this myosin head is on your thick filament. So you can think of this as a hand that's like pulling this rope inward toward the center. And then myosin uses ATP to return to its normal state, and then the cycle happens again. So basically what happens when your muscle contracts is this power stroke is happening, right? And so the basic unit of your sarcomere in a flat diagram looks more like this. So the 3D diagram is this, and so we said that sarcomeres are your basic contractile unit. So we weren't earlier looking at your sarcomeres, we were looking at a microscopic view of this area here. You could even see the myosin head a little bit there, and then how it would pull these inward. So I should probably draw it a little bit out for everyone. So if we go on here, make this bigger a little bit. So basically, if we have, so we were looking at a microscopic view here where you had the myosin head, and basically if it did the power stroke, it would pull this thin filament in. Just imagine this is your thin filaments here. This is your thin filament, all of these areas here. And your thick filament is right here, this big, thick area, as you can imagine, and it's all labeled for you. So you need to know the sarcomere, and it's even on your review sheet here. So you need to know the different parts. So this part right here is called the A band. This is where there's no overlap between your thin and thick filaments. It's just your thick. So that's called your H zone, right? And then your A band is your entire, I like to remember A for actin. So, and actin is your thick filament. So this is your entire A band, right? And then your I band is only where your thin filament is. I band. And then your Z disc is like the boundary between a sarcomere and another sarcomere. So they're repeating units, by the way. You don't just have one sarcomere, you have repeating. So Z disc is like the boundary between them. So yeah, we have all of that covered. So you need to know all of this part of your sarcomere, which is this diagram, which we just explained. Sarcomeres attach end to end to become microfibrils. Each myocyte contains many myofibrils, sarcoplasmic reticulum. So now we're entering a different kind of an anatomy to show you. So this is your sarcoplasmic reticulum, and you also have your sarcolemma and T tubules. So your sarcoplasmic reticulum is filled with a bunch of calcium. And we'll see later that calcium is really important for contraction, because we'll see later. And then your sarcolemma is the cell membrane of a myocyte, which is, let's see where we can find, there's the sarcoplasmic reticulum, where's the sarcolemma? Yeah, the sarcolemma is just the membrane of this thing, right? So this will probably have it, yeah, this membrane on the outside. and this is your entire muscle fiber, right? So that's just the membrane, and then your T-tubules are connected to the sarcolemma, and they carry signals. So this is actually really important that your T-tubules are carrying the signals. And what signals are they carrying? They're carrying they're carrying the electrical signal, or the, after an action potential fires, you need a firing of action potentials enough to carry through, and we'll see later why that's important, why you need action potential and the T-tubules to carry them through. So your T-tubules are there, and so when your muscle contracts, it begins at your neuromuscular junction. In other words, it's that area in between where the synapse happens. So remember, we're talking about neuron to neuron, and they have this gap, and in that gap, there's receptors on this side, and there's release of vesicles on the other side, right? So where efferent neuron release acetylcholine that binds to receptors. So acetylcholine is a neurotransmitter that gets released, and it binds to receptors on the sarcolemma. So the sarcolemma is the outside of the muscle, causing depolarization of the muscle membrane, or of the muscle cells. And that's why the T-tubules were so important, because they were bringing down that spread of electrical signal, and that triggers the release of calcium. That's why I said calcium was also important, because calcium will bind to troponin, which causes a shift. So we talked about earlier that troponin causes a shift in tropomyosin, which will lead to myosin binding sites with actin. So once myosin and actin bind, that's important, because then we can get our power stroke, right? So shortening of the sarcomere occurs. So that's basically the power stroke, because it brings in this together, making the whole unit shorter because of all the power strokes. And there's multiple myosin heads bringing it all together. This is the sliding filament model, or sliding filament theory. So this is important. If you don't understand it, type this in Google. So let me just get yellow color. Yeah, and the muscles relax when acetylcholine is degraded by an enzyme called acetylcholine esterase, terminating the signal, allowing calcium to return back to the sarcoplasmic reticulum. So, and then ATP binds back to the myosin head, as we saw earlier in the other diagram. There's not much diagrams about it here, so watching a video on the sliding filament model will probably help a lot too, allowing release from actin. So then the actin myosin binding site should release when the muscle is now resting and no longer in contraction, being contracted. A simple twitch is a single muscle fiber that responds to a brief stimulus. So that's only one twitch. But when you have frequent summation of a lot of like active action potentials at once, so much electricity or electrical signal happening, right? Multiple simple twitches before the muscle has a chance to fully relax, you get a frequency summation. So basically a sum of a lot of twitches. Oxygen debt is the difference between O2 needed and O2 present. Creatine phosphate adds a phosphate group to ADP, forming ATP. Myoglobin is heme containing protein that is muscular oxygen reserve. So I feel like this sounds very like separate, like just a bunch of facts, but you kind of want to relate it to real life scenarios somehow, and that will help you in the long run. And then lastly is the skeletal system that I left for last. So skeletal system is derived from your mesoderm. We already knew that from before. And so you have an axial skeleton, which involves your skull, vertebral column, rib cage, hyoid bone, and then your appendicular skeleton. So it's kind of like how we divide our central nervous system and peripheral nervous system. In the skeletal system, we have an axial and appendicular. And so the appendicular involves your bones of limbs, pectoral girdle, and your pelvis. And compact bone is very strong and dense. So you can think of your compact bone as like your thigh bone. That bone is probably really strong, right? And then you have spongy bone, which is a lattice-like structure of bony spinacles known as trabeculae, cavities filled with bone marrow. Bone marrow. Remember that bone marrow was important for a lot of cells that were forming and stuff. So yeah, bone marrow is very important. Bone marrow can be red. There's also yellow bone marrow. Yellow bone marrow has fat, and red bone marrow is filled with hematopoietic stem cells. So remember that's again where cell birth kind of happens. Long bones are shafts called diaphyses that flare to form metaphysis and terminate in epiphysis. So epiphyses contain epiphyseal growth plate. Peristonium is connective tissue that surrounds the bone. Ligaments attach bones to other bones. Tendons attach bones to muscles. And then bone matrix involves osteons, which are types of cells that is the structural unit of compact bone consisting of construction. So it has layers, lamellae, which surround the long hollow passageway, the Haversian canal, and between these rings are lunicae, where osteocytes reside, which are connected with cancaliculi. So for bone remodeling, osteoblasts will build the bone, and osteoclasts resorb bone. All right, and so this is pretty high yield, this part, because it's in a different color also because it's important. Parathyroid hormone increases reabsorption of bone and increases blood calcium levels. Vitamin D increases reabsorption of bone and increases blood calcium. Calcitonin increases blood formation, but decreases calcium in the blood. Cartilage. Cartilage is firm, elastic. Cartilage, like for example, is on your ear. It's like elastic part of your ear. Matrix is chondrin, secreted by chondrocytes, avascular, and is not innervated. So there's no like blood flow, for example, in that elastic part of your ear. Joints are immovable. Well, they can be. They're fused together to form sutures. They can be movable, strengthened by ligaments, and contain a synovial capsule. And then synovial fluid secreted by synovium lubricates the joints. And then for the fetus, bones form from cartilage through the endochondral ossification. Skull bones form directly from mesenchyme in the intramembranous ossification. So how about we quickly kind of find a picture for everything and then go on to chapter 12. Okay, I just wanted to take some time to show the compact and spongy bone and the osteon and the Haversian capsule. And also Volkmann canal is there as well. What else did I wanted to show? So I didn't really like this thing here. It actually confused me when I read it because I didn't learn this about osteoclast, that osteoclast resorb bone. I mean, yeah, I mean, it could be terminology wise, but I always learned osteoclast build bone, and then osteoblasts build new bone tissue while osteoclasts break down and dissolve old bone tissue. So this is a nice picture of basically what's happening. So yeah, I just want to go over that again. The matrix has a lot of just different names to memorize. So you have that osteons, lamellae, Haversian canal, osteocytes, and okay. So just a lot of pathways and names kind of thing. And that's about it. Yeah, joints are pretty easy, fetus. Okay, so everything is pretty good. Okay, and then I want to go over endochondral and mesenchyme. No, I want to go over endochondral and the other intramembranous ossification real quick. So just to give a quick overview, intramembranous ossification forms bone directly from embryonic connective tissue, which is also known as mesenchyme, without a template, building flat bones like skull and clavicle. But endochondral ossification replaces cartilage model with bone, creating most of the body's long and irregular bones. So yeah, that's basically a good description of that for this part here. Okay, now we're getting into the last chapter on genetics and evolution. I have some friends that love this part of genetics and stuff like that, and they studied science in that way because they loved it. We're almost done here. So definitions for genetics. So we'll go into definitions. So I talked about alleles before, that they're variants of a gene, or in other words, alternative forms of a gene. So an example could be blue eyes versus brown eyes versus light brown eyes. So there's a dominant allele, which for usually people with brown eyes, it's brown eyes, only requires one copy in order to be expressed. So it's so dominant, only requires one copy. So it would be like dominant, and then you can have a recessive allele. If you remember your Punnett squares, this is involved in it, but that's just one small section of an analytical technique as you see it here. Recessive allele requires two copies in order to be expressed, so you would have to have RR like this in order to be expressed. That's why it's recessive. That's a definition of it. Genotype, the combination of alleles has at a given locus. So if you're homozygous, having two of the same allele, so having two of the same allele could look like this, RR, that are both dominant, so brown eyes, or it could be something like this, RR, blue eyes, both recessive, blue eyes. And then heterozygous having two different alleles, which is like this, brown eyes plus blue eyes. Phenotype, the observable manifestation of the genotype. So your genotype can say you have a dominant R and little r, but your phenotype is going to be the dominant one, R, which in this example is brown eyes. So your phenotype is the observable manifestation of what shows up on the surface. Your dominance can be complete, codominance, or incomplete. If you have complete dominance, you have only one dominant allele. If you have codominance, then there's more than one dominant allele, and incomplete means there's no dominant alleles. So heterozygotes have intermediate phenotypes. Penetrance is when the proportion of individuals carrying a particular allele that also express an associated phenotype. Expressivity is the varying phenotype outcomes of the genotype. So just because it's brown eyes, it could be a little bit, different a little bit. Genetic leakage is flow of genes between species via hybrid offspring. So a donkey and a horse make a mule, I think. So hybrid, think about hybrids and stuff like that. Genetic drift is when the composition of the gene pool changes as a result to chance, just by chance. The founder effect is bottlenecks that suddenly isolate a small population, so it creates inbreeding. Breeding within that little small place. Taxonomic rank, a lot of people memorize it by this phrase here, King Philip came over from Great Spain, or you can memorize King Philip came over for great soup. I like the soup one better. I don't know why it's not there, because food is always good. So King, phylum, class, order, family, genus, species. So you just have to literally memorize that. And then before going into mutations, I'll probably go over Mendel laws. So Mendel's law has law of segregation and law of independent assortment. For law of segregation, an organism that has two alleles for each gene, which segregate during anaphase. So anaphase 1, remember that is part of meiosis, and because of this, gametes carry only one allele for a trait. So you only have one variant of a particular trait. You don't have two. So then, but this is for the gametes, because the gametes only have one. And then law of independent assortment is the inheritance of one allele does not influence the probability of inheriting a given allele for a different trait, except for linked genes. So I'll explain both of these in a second after we go through everything. So experiments is that people did experiments to test to figure out how genetics works, right? So they did some experiments to support that DNA was genetic material, that it was you could breed this way and this would happen. So Griffin demonstrated transformation, which heat killed smooth virulent strain of bacteria, still transformed rough strain into smooth. Avery, MacLeod, McCarthy, degradation of DNA led to secession of bacterial transformation. Degradation of proteins did not. So a lot of this is just explaining what happened, but I'm going to explain a little bit more actually. And Henrietta Chase confirmed DNA is genetic material because only radiolabeled DNA could be found in bacteriophage. So I'm going to explain this a little bit more simply. Alright, so we're looking at the Griffith experiment right now, a photo of it. And so it was done in 1928. And you can think of this as basically harmless bacteria which pick up a weapon from the dead and bad bacteria, so the virulent. So the smooth S strain is deadly/virulent. So as you see here, smooth, deadly virulent, and the rough R strain is harmless. So what Griffin did is he injected mice with heat-killed or dead S bacteria, so dead S bacteria. In other words, dead, smooth bacteria, so heat killed smooth strain. And what he found is that the mouse lived, but when he mixed harmless R bacteria, harmless R bacteria is right here, harmless R bacteria, and when he mixed it with the dead S bacteria, the mouse had died. So this mixing with the dead S bacteria still killed the mouse. So the takeaway from this is that something from the deadly bacteria, didn't know exactly what, but something from it passed into the harmless bacteria, right? This rough strain that was supposed to be non-virulent and gave them the ability to be deadly. As you could see here, the mouse died. And he called this mysterious passing of traits called transformation. So that's why we see on this, nope. That's why we see on this one, it's called transformed. Or transformation, demonstrated transformation. I'm going to highlight that word because it's important. And then now we're going to go into Avery MacLeod. So the Avery MacLeod and McCarthy experiment is very similar. And it's basically, they asked the question, what exact chemical inside of that dead bacteria? So they're basically building on top of Griffin. Like what inside of that dead bacteria was actually causing that transformation. So what they did was they took the mixture from Griffin's experiment and they used special enzymes to destroy one ingredient at a time. So the first thing they destroyed, from that mixture they destroyed proteins, and then the transformation still happened, right? So I didn't really like this one here. Maybe this one is better. So this one, so they took the mixture, the mysterious mystical mixture that Griffin found, and so they removed protein, and then they found that transformation still happened. And then they also tried to remove RNA, and they found transformation still happened. But when they removed DNA, they found that transformation had stopped completely. So the takeaway from that study was that they found out that DNA was the reason for the message to pass along, that dead bacteria that was supposed to be harmful to just be harmful because of DNA. So they found that DNA is genetic material that can be inherited. Hershey and Chase confirmed that DNA is genetic material because only radiolabeled DNA could be found in bacteriophage bacteria. So we'll go over that. So what happened was that I showed a picture of Hershey and Chase, their experiment. So some scientists were still very skeptical, and Hershey and Chase tested viruses, or also known as bacteriophages, that attack bacteria. And a virus is basically just a protein shell holding DNA inside of it. So remember we talked about bacteriophages, and they have bacteria inside of their capsule and stuff. So what they did was they used radioactive tags to make parts of this glow. So they made parts of it glow, and they tagged the protein code with radioactive sulfur to make a yellow glow, and they also made they also tagged the DNA with radioactive phosphorus to get a blue glow, right? And so what they found is that when the viruses attack the bacteria, only the radioactive DNA entered the bacteria into new viruses. So they would see colors, and then they would see actual things entering. And that was a cool part, because they figured out that, it was the DNA that was 100% being the genetic material that was transmitting over and passing down genetic instructions of what this cell should be doing. So their experiments were really good on defining what DNA was really doing. Okay, and then we have mutations that can happen with the DNA. So DNA is like a set of instructions, right? But sometimes those instructions are not made perfectly. So we have these mutations, and nucleotide mutations can be point mutations, frame shift mutations, and there's different results from those mutations that can occur. So point mutations is substituting one nucleotide for another. So DNA is made up of nucleotides. So here's a structure of DNA. Let me just show the nucleotides. So you have DNA with all their nucleotides, nitrogenous bases, adenine, thymine, guanine, and cytosine. You don't have to know this very much in biology, but in biochemistry, you will definitely know it by heart. And then so frame shift mutation is moving the 3-letter reading frame. So usually you read, oh, I hope that doesn't open up, 1 second. So this is the sequences. So adenine, guanine, and etc. just keep going in these strands, and basically people read them in 3. And basically a frame shift is if you like read them in 3, but moving the 3-letter reading frame over. Like you're moving it over. You're not substituting, but you're moving that reading frame over by 3 letters. And the results from these mutations could be silent, so you don't see any effect on the protein outcome. It could be missense, so you replace one amino acid for another, so the protein is composed of a different amino acid. And then nonsense result is when a stop codon replaces an amino acid. So instead of having an amino acid being coded for, it's just a period. Like that's it. No more code, no more reading frame, just a dot. That's it, stop. And then insertion deletion is a shift in the reading frame entirely, leading to a change in all the rest of the amino acids following it in the sequence. So you can imagine that insertion and deletion is the worst one, because then it's affecting the entire strain. Chromosomal mutations are much larger mutations, and they affect whole segments of DNA. So they can result in deletion, where a large segment of DNA is lost, duplication, where there's copies of the DNA, inversion, where the DNA is reversed, insertion, where a DNA is moved from one chromosome to another. There's translocation, where a segment of DNA is swapped with a segment of DNA from another chromosome. So there's chromosomal mutations, which are very big compared to only nucleotide mutations. So I just wanted to make it clear the difference between a nucleotide and a chromosome. So the nucleotide is a single chemical building block of the nucleic acids, like DNA, whereas the chromosome is a massive, highly packed structure. So you can imagine that if you have chromosomal DNA, it's going to be very problematic. So chromosome, again, was the organized package of long strands of DNA, tightly packaged around histones, which is like a protein, and then nucleotide is the microscopic monomer, the building block of DNA. So this is like microscopic little reading frames, not reading frames, but they're coded in reading frames, sorry. And then you have analytical techniques of genetics. Punnett squares is one of the ways you can deduce the probability of somebody's genotype and their phenotype. So the monohybrid cross accounts only for 1 gene. If you have a dihybrid cross, it accounts for 2 genes, and sex-linked cross is linked to the X chromosome. So remember that the X chromosome, so the mother carries all the information for like disorders and stuff like that, whereas the Y chromosome, not just the mother, but the Y chromosome carries only like information that you're a male. Stuff like it doesn't carry much information is what I'm trying to say. It's also smaller. Recombination frequency is the likelihood of 2 alleles being separated during crossover in meiosis. So the more farther apart, the higher, like the more likely. So the more likely the alleles will be separated if they're further apart during crossing over in meiosis. So the separation would happen in anaphase, right? And so the Hardy-Weinstein principle is that if a population meets a certain criteria aimed at a lack of evolution, then allele frequencies will remain constant. So it's possible, or that's his little principle here. And then he has an equation that kind of supports that. So P is equal to your dominant allele frequency, and then Q is your recessive, so P plus Q is equal to 1 whole. And then his equation is that P squared plus 2PQ plus Q squared is equal to 1. And then the evolution is that, actually 1 second, I'll explain that a little bit more. Alright, so what I wanted to show is that you can think of Hardy-Weinstein's equation. I have an analogy here of a jar of marbles. So imagine every person gets 2 marbles or alleles for their eye color. So they get brown marble and blue marble. So P is equal to your brown marble, which is your dominant allele, and your recessive is going to be blue, okay? So all the marbles in the whole population are then thrown into one jar. So all the marbles are equal to 1. And so in equation 1, since there are only 2 types of marbles in the jar, their percentages have to add up to 1, or in other words, 100%. Right, so if I multiply 1 by 100%, I get 100%. But if 70% of all the marbles in the jar are brown, then the remaining 30 would be blue. Would be blue. So if I have 70% people which have brown eyes, then that means 30% will have blue eyes, because that's 100% of the population. Okay, and then his second equation is about genotype frequency. So every person reaches into the jar and blindly picks up 2 marbles. Okay, so people are just picking up marbles blindly. So they're picking up either brown or blue eyes. And here there are only 3 possible combinations that a person can pull. So P squared is when the person picks up two dominant alleles, P and P. That's P squared. 2PQ is where you get a heterozygous P and P, like this, right? Or P and Q, in other words. And then and then you're adding them twice. And then Q is having a homozygous recessive, so Q and Q, so Q and Q. And basically, this is the combinations that someone can have, right? All the possible combinations will then equal 100% of the population, right? So this is kind of his equation of how his equation can be described, and this is how I described it. So evolution is natural. There's natural selection that can happen. The mechanism for evolution is natural selection. So people are naturally selecting their partners, right? Modern synthesis model is neo-Darwinism. Mutation and recombination are mechanisms of variation, differential reproduction. And then inclusiveness. If a population meets a certain criteria aimed at a lack of evolution, then allele frequencies will remain constant. So, and then punctuated equilibrium considers evolution to be a very slow process with intermittent rapid bursts. So it's sometimes have these like random bursts of randomness and then change, right? Mode of natural selection. So there can be a stabilizing selection which keeps phenotypes in a narrow range. So only people with blue eyes kind of thing. Directional selection, it moves in the average phenotype toward an extreme. So the average is gonna be like, let's say the average was all brown eyes and then the extreme would be blue eyes kind of thing. Disruption selection moves towards two different phenotypes at the extremes, can lead to speciation. So I'll highlight some vocab words and stuff like that. I'll describe some of them again, just because they're a little bit, they should be explained better. Adaptive radiation is a rapid emergence of multiple species from a common ancestor. Each has a niche. Isolation is reproductively isolated from each other by pre or post-zygotic mechanisms, and the molecular clock models the degree of difference in the genome between two species and is related to the amount of time since the two species broke off from the common ancestor. So I'm going to explain a little bit more of all of these with the context toward it. So in your models, modern synthesis model, which was the neo-Darwinism, right? The concept of Charles Darwin came up with natural selection, which is over here, but he didn't talk about DNA or genes. So what the modern synthesis model is looking at is simply Darwin's theory, but it's updated with a way to include our modern approach with genes and DNA and stuff like that. So how it works is that random DNA mutations would then mix our population. So differential reproduction, mutation and recombinations are mechanisms of variation. So in other words, the reason why we have variation in the population is because of mutations and stuff like that. And then differential reproduction, individuals with helpful traits survive and have more babies and they pass those genes on. So that's what differential reproduction is. And then for inclusive fitness, what it really means is success isn't just about like you having kids, it's also about helping the family members then survive. So that is what inclusive fitness basically is. And then for punctuated equilibrium, the concept is that evolution doesn't always move steadily. Instead, it moves in constant speed. It moves in constant speed. So let's say you sit slow in a traffic stop for hours, but then, later on the road clears and now it's all good. And then you have a short rapid burst of change. And then you have 3 modes of evolution. So for the stabilizing one, right, an example would be human birth rate. So babies that are too small or too large, they face higher health risks. So then medium weight babies survive best. So that's why the average would work the best in this situation. And then for directional selection, it's basically like a giraffe's neck. Their necks are so tall because over time, what happened in evolution, trees grew taller, so only giraffes with longer necks could survive. So it went to the extreme where they grew really long necks. So the whole population shifted toward that extreme. And then disruptive selection is where you have both extremes that dislike the middle. So you only have either, tall necks or really short necks, for example. So some birds have really tiny beaks, for example, and some birds have huge beaks. But maybe in an island that only had tiny seeds or giant nuts, you'd only have birds that have either giant beaks or small beaks because there's only small seeds or giant nuts to eat. So then the medium-sized birds wouldn't do well in survival and they would die off, essentially. So disruptive selection is when you get two extremes. And then adaptive radiation is basically the concept that one common ancestor rapidly branches off into many different species to fill empty ecological jobs, which are called niches. So in everyday analogy is that a food court opens with 5 empty restaurant stalls and one chef arrives and opens a pizza place, a sushi spot, a burger joint, and a salad bar to satisfy every different craving. So basically one common ancestor, that one person decided to branch off into different niches, like different food types, for example. And then the classic like biology examples like Darwin's finches branching out into different beak shapes to eat depending on the available foods that there were. So that's adaptive radiation. So coming from a common ancestor is key about that one. And then for reproductive isolation, for one species to officially split into 2, they must stop breeding together. So by pre or post-zygotic mechanisms. So pre-zygotic is before fertilization, something stops them from mating in the first place, right? And post-zygotic, which is after fertilization, is that they can mate, but the resulting offspring cannot continue the lineage any further. So an example of that is a horse and a donkey. They can produce a male, but the male is sterile, so they can't produce any further and continue that lineage. So that's how isolation can occur. An example of pre-zygotic, so before fertilization, is that if you live in different habitats completely and you mate at different times of the year or have different like mate dances or anything that stops two species from mating in general, anything really. Molecular clock model is basically that like random or neutral DNA mutations accumulate at relatively steady constant rate over time. So the degree of difference in the genome between two species related to the amount of time since the two species has broken off from the ancestor. So if you count the number of DNA differences between the two species, if two species have very few differences, that means they split apart just recently. For example, humans and chimpanzees or chimps, right? And let's say you have two species that have many differences. So that means their common ancestor split apart a very long time ago. For example, humans and fish, they're very different. So their common ancestor would have been like long gone from now, right? So that's the molecular clock model. And that's about it. That's literally all of biology there.